There is a small pink animal living in a glass tank in a child's bedroom in Manchester right now that can regrow its own spine. Not metaphorically, not in some loose inspirational sense. The animal in that tank, assuming the tank is properly cooled and the water is changed on schedule, and the child remembered to feed it this morning, has, encoded in its body, the ability to regenerate vertebral column, spinal cord tissue, and the neurons running through it. If the tail were severed at this moment, in roughly 40 days the tail would be back. Not a stub, not a scar. The tail, bone, muscle, nerve, the lateral fin, the fine pattern of the skin, functional, indistinguishable from the original to anyone who hadn't watched it happen. The animal, of course, is an axolotl, and you have almost certainly seen one, probably more than once. If you've played Minecraft in the last 5 years, you've collected one in a bucket. If you've played Pokemon, you've met its cartoon descendants, Wooper in 1999 and Mudkip in 2002, both designed in homage to this specific salamander. If you've spent any time on TikTok, the algorithm has at some point served you a video of a softly smiling pink creature in a small aquarium, gills fanned out around its head like a feathered crown, eyes fixed on nothing in particular. If you've held a Mexican 50 peso note, you've held an axolotl. It's printed on the back, alongside maize and the Chinampa wetlands of Mexico City, where the animal evolved. This is the animal you know, the pet store axolotl, the internet axolotl, the smiling one. It is also, in some important and slightly disorienting sense, not quite the real animal, the real one. The one that evolution actually produced, the one that has lived in the lakes beneath Mexico City for somewhere between 10 and 15 million years, is dark olive brown with faint gold flecks, almost invisible against silt. It is also, at the time of this recording, very nearly extinct in the wild. The most recent IUCN assessment puts the entire wild adult population somewhere between 50 and 1,000 individuals. The most recent population modeling out of the National Autonomous University of Mexico suggests that, on current trends, the canals of Xochimilco may not contain a single wild axolotl by the end of next year. There are, at the same time, hundreds of thousands of axolotls alive on Earth, possibly millions. Almost all of them sit in tanks, in laboratories, in living rooms, in classrooms, in pet shops in Tokyo and Berlin and Dallas and Manchester. Almost all of them, traced back through their lineage, descend from a shipment of 34 salamanders that arrived in Paris by ship in 1864 and were placed in the care of a French zoologist named Auguste Dumeril, who initially believed, on the authority of Georges Cuvier, that the animals were the larvae of some unknown adult species. Within a year of the shipment's arrival, the larvae began laying eggs, and Dumeril realized that Cuvier had been wrong for 50 years. This is part of the strangeness. The axolotl is everywhere and almost nowhere. It is the most photographed salamander on the planet and the most endangered amphibian in Mexico. It carries in its cells the most complete known regenerative program in any vertebrate. It has 32 billion base pairs of DNA, 10 times the human genome, and biologists do not yet fully understand what most of that genome is for. It refuses to grow up. The Aztecs noticed this 900 years ago and built a god out of it, a god whose name Xochimilco the salamander still carries, a god who, in the oldest version of the myth, refused his own sacrifice and fled into the water, transforming himself into the animal that would not transform. The Aztecs, working without microscopes, came surprisingly close to a fact that modern biology only confirmed in the 20th century. That this particular salamander had refused, somewhere far back in its evolutionary past, to complete the standard amphibian transition from water to land. And that this refusal, this decision, if such a word fits, is the source of nearly everything astonishing about it. the reason it can rebuild its own limbs, the reason its cells can step backward into something like an embryonic state and then forward again, the reason it does not develop tumors at the rates a hard-working dividing cell animal ought to develop them. All of this appears to be tied, in ways researchers are still working out, to the simple fact that the axolotl never fully grows up. There is more, and most of it is stranger than the parts already mentioned. But this is the animal. The pink one in the tank, the dark one in the canal, the one Dumeril stared at in 1865, and the one James Monaghan's lab in Boston is currently running through CRISPR experiments. The same animal. Stretched between extinction in the wild and proliferation in captivity, between Aztec mythology and regenerative medicine, between a child's bedroom and a UNESCO wetland that may not survive another decade. If you'd like to hear how all of that fits together, how the science actually works, how the animal got here, what it means that it carries inside itself a partial answer to questions about human healing that medicine has been asking for 2,000 years, settle in. The answers take some time. They are worth the time. A small favor before we begin. If something in the next 2 hours feels worth being made, the subscribe button helps a channel like this one more than almost anything else does. And in the comments, I'd be curious to know, when did you first encounter an axolotl and where? A pet shop, a video game, a textbook, a museum, a bedtime story? I read the answers, and they tell me something the data can't. Somewhere tonight, in the canals beneath one of the largest cities on Earth, a dark salamander is hovering motionless above silt, gills barely moving, watching the water for the slow chemical signature of a worm. It does not know it is endangered. It does not know it is famous. It does not know that its body, its specific evolutionary inheritance, is being studied in laboratories on 3 continents because of what its cells can do. It is just an animal in a place doing what it has done for millions of years. Good night for now. The story begins in 1500. Tenochtitlan, sometime in the early 1500s, an hour before dawn. The lake is flat and dark and smells of damp reeds and the wood smoke of cooking fires that have not yet been relit. Somewhere out on the canal, a heron calls once and goes quiet. A man in a long cotton mantle is standing knee-deep in the shallow edge of a chinampa, one of the rectangular farming islands the Aztecs have engineered out of the lakebed, holding a small woven net. He is hunting axolotls for the festival of Izcalli, which begins in a few days, and he needs perhaps a dozen of them. They will be steamed inside maize husk parcels with yellow chilies and salt and given as offerings to the old fire god of the hearth. The Florentine Codex, which a Franciscan friar named Bernardino de Sahagún will compile 60 or 70 years from now, will describe the dish in 3 words that translate roughly as good, fine, savory. The man reaches into the water with a slow hand. He is not in a hurry. The animals he is looking for are dark olive green with faint gold flecks along the spine, and at this hour, in this light, they are nearly invisible against the silt. He has been doing this since he was a boy, as his father had, and his father's father, and a very long line of fathers before that. People have been pulling these particular salamanders out of these particular lakes for at least 8,000 years. There are middens around the basin of Mexico filled with their tiny, unmistakable jawbones. The animal he is hunting, the people of Tenochtitlan call axolotl. The word is built from 2 Nahuatl roots that the language layers together with characteristic density. Atl means water. Xolotl is more difficult. Translators have rendered it as dog, as monster, as servant, as deformed thing, as twin, and none of those is quite right because the word holds all of them at once. Axolotl, then. Water dog, water monster, water twin. a thing of the water that does not entirely belong to the water. It is also the name of a god. In the religious imagination of the Aztecs, Xochimilco was the dark twin of Quetzalcoatl, the feathered serpent. Where Quetzalcoatl was depicted as bright and ascending, the morning aspect of the planet Venus, Xochimilco was depicted as canine-headed, stooped, with reversed feet, the evening aspect of the same planet. He was the god of fire and lightning, of twins and monsters and deformities, and his particular ritual job was to guide the dying down into Mictlan, the Aztec underworld. When a person died, a small dog was sometimes sacrificed and buried with them, and the dog, in some sense that was both literal and metaphorical, was Xochimilco, walking ahead with the soul into the dark. There is a story about him, recorded by Sahagún and others, that sits at the center of the Aztec creation cycle. It runs roughly like this. The world has just been remade after several previous worlds have ended in catastrophe, and the gods are gathered to set the new sun and moon in motion. To do this, sacrifices are required. Several gods have already given themselves. One more is needed to make the sun actually move across the sky, and Xochimilco, who has watched the others go before him, refuses. He does not want to die. He flees the place of sacrifice. The other gods hunt him. He hides among the maize stalks in a field, transforming himself into a stalk of corn with 2 ears, the kind farmers call Xochimilco corn even today, when one stalk produces 2 cobs. The hunters find him. He flees again and hides in a field of maguey, the agave plant, and transforms into a doubled maguey, a mexolotl, 2 plants growing as one. The hunters find him there too, so he runs to the water and dives in and transforms into the animal that will from then on carry his name. The hunters catch him eventually, and the sacrifice is completed and the sun begins to move. But the form he chose last, the form he chose to refuse death in, is the salamander. The animal in the canal is a god who refused to grow up. This is the part that is genuinely strange, and it is worth pausing over before continuing, because what the Aztecs were describing in mythological terms turns out to map with uncomfortable precision onto a fact about the animal that European biology would not arrive at for another 350 years. The axolotl, alone among the salamanders of the central Mexican highlands, does not metamorphose. It does not, under normal conditions, lose its gills, develop lungs, climb out of the water, and live as an adult amphibian on land. It stays in the larval form for its entire life. It reaches sexual maturity, reproduces, ages and dies, all while retaining the body plan of a juvenile. It refuses, in the technical biological sense of the word, to mature. The Aztecs did not have access to thyroid hormones or developmental genetics or the comparative anatomy of urodele amphibians. They watched the animal for centuries and built a story about a god who would not accept the ordinary fate of his kind, who would rather become a strange small creature in the lake than complete the transformation expected of him. The story is, as a piece of biology, almost embarrassingly accurate. Whether this is coincidence or whether it reflects something the Aztec cosmologists genuinely observed and encoded, that this animal seemed somehow to be a kind of frozen child of its own family, never quite finished, is impossible to say from this distance. The cosmology came first, and the cosmology fits the biology too well to ignore. What can be said is this. By the time the Spanish arrived in the 16th century, the axolotl had been part of Mexican religious, agricultural and culinary life for, conservatively, the better part of a millennium. It appeared in the codices as a deity. It appeared in the markets as a meal. It appeared, ground into pastes and infusions, in the medicine of native physicians who associated it with healing, particularly according to the records assembled by the Spanish royal physician Francisco Hernandez in the 1570s, with conditions involving wasting and weakness. The Aztec attribution of regenerative power to the animal was, again, not exactly wrong. We will come back to that. Hernandez, working under commission from King Philip II, spent 7 years in New Spain documenting the medicinal plants and animals of the region, interviewing indigenous physicians, and producing a manuscript of staggering size, 16 volumes of natural history, much of which would be lost in a fire at the Royal Palace in Madrid in 1671. What survives includes a brief, careful entry on the axolotl, the first scientific description of the animal in any European language. Hernandez described it accurately, did not understand what it was, and noted its unusual features, the external gills, the strange persistence of larval characters, without committing to a classification. He sent his manuscript home. The fire took most of it. The animal stayed in its lake. For the next 2 centuries, almost nothing happened. The Spanish drained the upper lakes of the Basin of Mexico, cut the canals, built churches over the chinampas, and reorganized the entire water system of the valley to suit a colonial city. Axolotls continued to live in what was left, and continued to be eaten and continued to be sold in local markets, but they did not enter European scientific awareness in any serious way. The first major European scientist to take an interest in them would not arrive in Mexico until 1803, when Alexander von Humboldt, the Prussian naturalist whose name now attaches to ocean currents and squid and several mountain ranges, walked through the markets of Mexico City and saw on a vendor's table 2 small dark salamanders that he had never seen anything quite like before. He bought them. He preserved them in alcohol. He carried them back to Paris in a glass jar, along with several thousand other specimens, and gave them to the most authoritative comparative anatomist in Europe, a man named Georges Cuvier, who looked at them carefully, considered the question of what kind of animal they were, and arrived at an answer that turned out to be wrong in an interesting and revealing way. The mistake would last 50 years. It would only be corrected by accident by a man whose father had also been wrong about the animal, in a converted monkey house at the edge of the Jardin des Plantes on a spring afternoon in 1865. But the lake the man on the Chinampa is standing in, the lake the heron just called over, has another 4 centuries of survival left in it before that story matters. For now, the man pulls his net up out of the water with 2 dark animals in it. He drops them into a clay jar. He moves to the next stand of reeds. The sun has not yet come up. The festival is in a few days. The salamander in the jar does not know it is a god. It does not know that the Spanish are coming. It does not know that in 500 years, almost every member of its species will be living in glass tanks in cities its ancestors could not have imagined, and that its wild descendants, the dark animals in the silt of these particular canals, will be down to a few hundred individuals, hovering on the edge of disappearance. It only knows the inside of the jar. The man carries it home along the Chinampa edge, the canal water still moving slightly in the dim light, the city beginning to wake around him. The animal will be cooked in a few days. Other animals just like it are continuing their mourning under the surface of the lake, motionless, waiting for the slow vibration of a worm. The 2 preserved salamanders that Alexander von Humboldt brought back from Mexico arrived at the Muséum National d'Histoire Naturelle in Paris sometime in 1804, packed in alcohol in a small glass jar that had crossed the Atlantic in the hold of a ship, along with several thousand other specimens, pressed plants, mineral samples, beetles in cotton wool, a great many dried birds. The jar was unremarkable. The animals inside it were dark, soft-bodied, and a little under 20 centimeters long. Their gills were still attached. They had been dead for somewhere between several months and a year. Humboldt had collected them in the markets of Mexico City, where the local people sold them as food, and he had been told a great deal about them by his Mexican hosts, including the fact that they retained these strange external gills throughout adult life and reproduced in that condition. He passed the jar to the man best qualified to identify it, Georges Cuvier. Cuvier, at this point in his career, was the most authoritative comparative anatomist in Europe and arguably the most powerful naturalist in the world. He had built his reputation on the principle that an animal's anatomy is internally consistent, that, given a single bone, an experienced eye could reconstruct the rest of the skeleton, the muscles, the diet, the habitat, even the likely behavior of the creature it came from. He had used this principle to dazzling effect in his work on fossil mammals, identifying extinct species from fragments and establishing, almost single-handedly, the science of paleontology. What Cuvier brought to a problem was structure. He looked at an animal and asked what kind of body plan it represented, what category it fit into, what the underlying logic of its parts was telling him. He looked at the two salamanders from Mexico, and he saw something his comparative method had taught him to recognize. He saw a larva. The reasoning given his framework was almost airtight. Adult amphibians in the European fauna Cuvier knew best lose their gills. They develop lungs. They climb out of the water and live, at least for part of the year, on land. The presence of external gills in a vertebrate animal of this size and shape was, to Cuvier, a clear anatomical signal that the creature in front of him had not yet completed its development. It was a juvenile. The adult form, whatever it was, must exist somewhere in Mexico, presumably as a larger lung-breathing terrestrial salamander that had simply not been collected yet, or had been collected and not recognized as the same species. Cuvier wrote up his judgment, included it in his great 1817 work, Le règne animal, and moved on to other questions. There were a few naturalists, mostly working in Germany, who suspected he was wrong. Reports kept arriving from Mexico that the animal lived its entire life in the water, that adult specimens had been seen with gills, that the indigenous fishermen who had been catching axolotls for thousands of years had no concept of the animal as a juvenile of anything else. Cuvier read these reports. They did not move him. In the second edition of Le règne animal in 1829, he addressed the problem with a kind of weary frustration. He acknowledged that so many witnesses, he wrote, assured him the gills were not lost that he was obliged to classify the animal among the permanent gill breathers. But he made it clear in the same passage that he found this conclusion implausible, that his own reading of the anatomy still suggested a larval form, and that he was deferring to testimony rather than to comparative reason. The phrasing is the phrasing of a man bowing reluctantly to a fact he does not believe. He died in 1832. The question stayed open. For another 32 years, European zoology more or less accepted Cuvier's reluctant verdict. The axolotl was a strange permanent larva, a member of some new and somewhat unsatisfactory amphibian category, and got on with other things. Various names were proposed. The German zoologist Johann Wagler put it in a genus of its own, Cirridon. The anatomy was diagrammed. The animal stayed, in scientific terms, frozen in its categorical confusion, suspected of being a larva, formally classified as something else, and not actually present in any European laboratory in living form, for anyone to settle the question by direct observation. That changed in 1864. In the autumn of that year, a French scientific expedition that had been sent to Mexico under the auspices of Napoleon III's brief and disastrous imperial adventure in the country, the expedition that placed the Habsburg Archduke Maximilian briefly on a Mexican throne, was wrapping up its biological collecting. One of the naturalists attached to it, a man named Marie-Firmin Beaucourt, had been instructed to bring back living specimens of the more interesting Mexican fauna. Among the animals he packed for the return voyage were 34 axolotls taken from the canals of Xochimilco and Lake Chalco, sealed into containers of fresh water and loaded onto a ship at Veracruz. They survived the crossing. All 34. This is, even by the standards of 19th-century animal transport, remarkable. The mortality rate for living amphibians on transatlantic voyages was generally close to total. The animals arrived in Paris in the autumn of 1864 and were divided between two institutions. 6 of them went to the reptile collection of the Muséum d'Histoire Naturelle, which was housed in a converted monkey house at the edge of the Menagerie, the museum's small and unfashionable zoological garden. The remaining 28 went to the more public-facing Jardin d'Acclimatation, an animal acclimatization society that had been set up in the 1850s to introduce exotic species to French zoos and farms. The 6 that went to the museum were placed under the care of the curator of reptiles and amphibians, a 52-year-old herpetologist named Auguste Dumeril. Dumeril was the son of Constant Dumeril, the previous curator, who had personally examined Humboldt's preserved specimens with Cuvier 60 years earlier and had agreed with Cuvier's larval interpretation. Auguste had grown up around these animals as a question. His father's question, Cuvier's question, the question of what the axolotl actually was. He inherited the curatorship in 1857, and when the 6 living animals arrived in his care in 1864, he began, more or less immediately, to do what zoologists do with new acquisitions. He tried to keep them alive, observe them, and figure out what they were. His first formal report on them, dated the 31st of October, 1864, is brief and unremarkable. He described the animals' appearance, noted their continuing gills, and confirmed in a single sentence his father's and Cuvier's classification. They were, he wrote, larvae. 6 months later, he had to take that back. The animals had reproduced. In the spring of 1865, in the converted monkey house at the edge of the Jardin des Plantes, the female axolotls began to lay eggs. The males fertilized them in the species' characteristic way. The courtship dance, the deposited spermatophore, the female collecting the sperm packet with her cloaca. The eggs developed normally. They hatched into larvae. The larvae, in due course, grew into adult axolotls indistinguishable from their parents, gills and all. None of the parents had metamorphosed. None of them had ever shown any sign of becoming something else. They had simply, while retaining every characteristic Cuvier had identified as larval, behaved as adults, which is to say they had reproduced, and reproduced fertilely, and produced viable offspring of the same form. This is not, in biology, an ambiguous result. Larvae do not reproduce. The category is defined in the comparative anatomy tradition Cuvier himself had built by the fact that the larval form precedes sexual maturity. Whatever is sexually mature, by definition, is no longer a larva. If these animals were laying viable eggs, they were adults. And if they were adults, then adults of this species kept their gills, lived in water, and never metamorphosed, which meant Cuvier had been wrong. Constant Dumeril had been wrong. 50 years of European amphibian classification had been wrong in a way that had been hidden not by lack of evidence, but by lack of access to a living animal under conditions where it could simply be allowed to grow up and do what its biology made it do. Auguste Dumeril reported the breeding to the Académie des Sciences in the summer of 1865 in language that is, for a French scientific report of the period, almost humble. He described what had happened. He noted that the result raised, as he put it, uncertainties regarding the true nature of these batrachians. He did not gloat. He did not particularly defend his father or attack Cuvier or claim any priority for himself. He simply reported the eggs, the larvae, the adult forms, and the obvious conclusion. The animal was not a larva. It was an adult in larval form, which meant the European framework for understanding amphibian development needed adjustment. The framework eventually made the adjustment. The category that emerged from this, neoteny, the retention of larval features in a sexually mature animal, would become, over the following century, one of the most important concepts in evolutionary developmental biology, with implications reaching far beyond salamanders and far beyond the question of axolotls in particular. But all of that was downstream. In 1865, Dumeril simply had a converted monkey house full of breeding salamanders that should not, by the lights of the most authoritative anatomist of the previous generation, have been able to breed. He kept them. He bred them. By 1866, the colony numbered 800 individuals. He distributed them generously and almost casually to other European laboratories, to Berlin, to Vienna, to Leipzig, to London, wherever a colleague wrote and asked for a few. By the early 1870s, there were axolotl colonies in essentially every major zoological institution in Europe, and the animals were beginning to be sold to private aquarium hobbyists, of whom there were a surprising number in mid-Victorian Europe. The species, which had been confined to a single lake system in central Mexico for the entirety of its evolutionary history, was now distributed in the form of laboratory and aquarium populations across an entire continent. Nearly every one of those animals could be traced, in genealogical terms, back to the 6 that had been placed in Dumeril's care in 1864 and the cohorts that arrived shortly after. Dumeril himself did not see most of this. In 1867, he published 2 short papers reporting something he had noticed almost in passing while caring for the colony: that the animals, when they damaged themselves in the holding tanks, regrew the damaged parts. Bitten gills came back, lost toes regenerated, tails, severed in fights between males during the breeding season, were reconstructed over the following weeks. He wrote up these observations in the manner of a man describing a curiosity. He did not have the conceptual framework to recognize what he was looking at. The papers are now generally considered the founding documents of modern regenerative biology, but in 1867 they read as natural history notes from a careful observer who had stumbled across something he could describe but not yet explain. He was busy with other things: the colony, the museum's collections, the wider work of his curatorship. In September of 1870, the Prussian army surrounded Paris. The siege lasted 4 months. Dumeril, who was 62 and not in good health, did not survive it. He died in early 1871 in a city where the bread lines were measured in hours and the axolotl colony in the converted monkey house was being kept alive on whatever food the museum staff could find for it. The colony itself survived. Some of the animals were eaten during the siege by museum staff who had run out of options. The rest were maintained, fed, and eventually expanded by Dumeril's successors. The animals he had received in 1864 are, in a real and traceable sense, the ancestors of the laboratory axolotls now living in Vienna and Boston and Lexington and 100 other research institutions. The line is unbroken. The Paris colony went out in shipments across Europe. From Europe it went eventually to North America. from a single 1935 import of 5 animals into the United States, a man named Rufus Humphrey built the colony at the University of Buffalo, which moved to Indiana in 1957 and to Kentucky in 2005, where it now constitutes the Ambystoma Genetic Stock Center, the central reference population for axolotl research worldwide. Almost every animal in that population and almost every captive axolotl on Earth descends from the 34 salamanders that crossed the Atlantic on a French ship in 1864 and the 6 that ended up under Auguste Duméril's care. Cuvier had been wrong about the animal for 50 years. He had been wrong because the framework he was using to think about amphibian anatomy was, in this one specific case, betraying him. Telling him to see a larva when there was no larva, telling him to expect a metamorphosis that would never come. The animal had been quietly being itself the entire time, in the lakes south of Mexico City, holding its strange position outside the standard amphibian life cycle, neither juvenile nor exactly adult by the categories of European biology, just continuing. It took the simple act of bringing it to Paris alive and letting it breed in a converted monkey house to undo half a century of misclassification. The animal had not changed, the framework had to. In the canals of Xochimilco, meanwhile, while all of this was happening in Paris, the wild population continued. The Spanish drainage projects of the previous 3 centuries had already begun to shrink the original lake system, but the reduction was not yet catastrophic. Xochimilco itself remained, more or less, as it had been under the Aztecs, a network of canals between chinampas fed by springs used for agriculture and transport, full of dark salamanders moving slowly through silt. The major decline was still 100 years away. The animal in the lake was, for the moment, simply present, in the way it had been present for the previous 10 or 15 million years, doing what its biology made it do, indifferent to the European debate about what it was. What its biology made it do, however, turned out to be considerably stranger than even Dumeril had realized. The retention of gills, the failure to metamorphose, the breeding in larval form, all of these were unusual, but they were the visible part of the animal. The invisible part, the part that lay inside its cells and tissues and wound-healing responses, would take another century and a half of laboratory work to begin to understand, and the work would begin almost as a footnote with the observations Dumeril made in 1867 about regrown gills and reconstituted tails. He had no idea what he was looking at. Almost nobody, for nearly 100 years afterwards, had any idea what they were looking at. The animal kept its secret in plain sight, in tanks all over Europe, while biology slowly developed the tools to ask the right questions about it. Those tools, when they finally arrived in the 20th century, would reveal something for which Cuvier's framework had no category at all. Not a larva, not exactly an adult, something stranger than either, doing things to its own body that no other vertebrate of comparable complexity had ever been observed to do. There is an animal sitting in a research tank in a laboratory at the Institute of Molecular Pathology in Vienna late in the evening, and a graduate student has just lowered the lights for the night. The water in the tank is held at a steady 15 degrees Celsius, a few degrees cooler than tap water, a few degrees warmer than refrigerated milk, because that is the temperature range in which axolotl cells appear to function most cleanly, and the cooling unit hums quietly in the corner of the room. The animal is about 20 centimeters long. It is not moving. It is doing what axolotls do for most of any given hour, which is hovering motionless about a centimeter above the gravel, gills fanned slightly outward, its broad, flat head pointing toward nothing in particular. To a casual observer, the animal looks asleep, or bored, or simply inert. None of those descriptions is correct. The animal is paying attention. in several different ways at once to information that does not reach a human nervous system at all. If you were to walk up to the tank and place your hand against the glass, the animal would know. Not because it would see you, its eyesight is poor, lidless, designed for detecting changes in light intensity rather than for resolving shapes, but because the slight pressure of your palm on the glass would deform the water inside the tank, and that deformation would propagate to the animal as a faint mechanical wave. Along the sides of its body, in two parallel lines that run from the back of the head to the base of the tail, there are small clusters of receptor cells called neuromasts, each one capped with a tiny gelatinous cupula that bends in response to water movement. The system is called the lateral line, and most of the world's fish have one. Axolotls have an unusually well-developed version. Those neuromasts can register water displacement on the order of micrometers, which means that the animal can feel, quite precisely, where things are around it, how big those things are, how fast they are moving, and in what direction. A tilapia drifting past in a Mexican canal at night is not, to an axolotl, a thing it has to see. It is a pattern of pressure waves moving across the side of its body, and those waves carry, in their shape and timing, almost everything the animal needs to know about the intruder. There is more. Clustered around the same regions of the head, particularly along the base of the external gills and the lower jaw, the axolotl carries a second kind of receptor, smaller, more numerous, and considerably stranger in its function. These are called ampullary organs, and they detect electric fields. Every living thing in water generates a faint electrical signature, muscles firing, nerves discharging. Even the simple movement of charged ions across a fish's gill membranes produces a measurable field in the surrounding water. Sharks famously have ampullary organs, the so-called ampullae of Lorenzini, which they use to find prey buried in sand or hidden in murky water. Most people are surprised to learn that salamanders have them too. The axolotl does. Sitting at the bottom of its tank, motionless, gills barely moving, the animal is reading the electrical fingerprint of every other living creature within a meter or so of itself. A small crustacean buried in the gravel does not have to move to be detected. Its heartbeat is enough. This combination, strong lateral line, functional electroreception, an excellent sense of smell delivered through paired olfactory organs in the snout, and very poor eyesight, tells you almost everything you need to know about how the animal hunts and where it evolved. The lakes of the central Mexican highlands, particularly the canal system at Xochimilco, are not visually clear environments. They are rich in suspended sediment, organic detritus, decaying plant matter, and the runoff of whatever lives upstream. For most of the day, the water is the color of weak tea. At night, it is the color of nothing at all. An animal that hunts by sight in these conditions will starve. An animal that hunts by pressure, smell, and electric field will not. What it hunts mostly is small invertebrates, aquatic worms, insect larvae, freshwater shrimp, amphipods, the occasional small fish or tadpole that strays too close. Axolotls are also, given the chance, willing to eat each other, particularly each other's external gills and digits, which they bite off in territorial disputes and during competitive feeding, and which their wounded conspecifics promptly grow back. This last point will become important. The hunting itself is not a chase, it is a wait, followed by a single explosive movement, followed by another long wait. An axolotl picks a spot, typically near a stand of vegetation or some kind of cover, and stays there. It barely moves. Its breathing slows. The gills, which look so dramatic in a photograph, are at this moment pulled slightly inward and held still. The animal is a piece of the substrate. When something edible drifts within range, The axolotl does not lunge in the way a fish lunges. Instead, in something like 20 milliseconds, about a fifth of the time it takes a human to blink, it opens its broad jaw and creates a sudden vacuum inside the mouth cavity, and the prey is pulled in along with several milliliters of water. The teeth are small and pedicellate, designed not for cutting or chewing but for gripping. The animal swallows its prey essentially whole. The whole sequence is over before a human eye can register that anything has happened. The axolotl returns to motionless. Whatever was alive a moment ago is now in its stomach. The body that performs this trick is unusual in several ways that are worth slowing down to look at. Internally, the axolotl has a three-chambered heart of the kind common to amphibians, which is to say 2 atria and a single ventricle that mixes oxygenated and deoxygenated blood somewhat inefficiently. It has lungs, small, undeveloped, but functional, and on rare occasions, particularly when the water in its environment becomes oxygen poor, an axolotl will drift to the surface, take a quick gulp of air, and return to the bottom. It also breathes through its skin. Most amphibians do this to some extent, but the axolotl does it to an unusual degree, supplementing both the gills and the lungs with cutaneous gas exchange across its entire body surface. The arrangement looks redundant on paper, 3 respiratory systems for a small aquatic animal, but it makes sense if you consider what the animal evolved to live in. Xochimilco is not a stable oxygen environment. The water warms in the dry season, cools in the wet, runs muddy after storms, and has had its hydrology actively engineered by humans for the better part of 2,000 years. An animal with 3 independent ways of getting oxygen out of its surroundings can tolerate conditions that a single system animal cannot. Redundancy for the axolotl is not extravagance, it is insurance. Externally, the most striking feature is the gills. 3 pairs of them branching out from either side of the head like fronds, lined with thousands of fine vascular filaments, the structures called fimbriae, that maximize surface area for gas exchange. The gills are not static. The animal flicks them, pulses them, holds them in different positions depending on what it is doing. When the water is rich in oxygen and the animal is at rest, the fimbriae lie flat and the blood inside them moves slowly. When the water becomes hypoxic, or when the animal is digesting a meal and needs more oxygen than usual, the gills flare wide and the blood flow accelerates, sometimes visibly. A relaxed axolotl has gills that look almost ornamental. A working axolotl has gills that look biological, full, dark, pulsing with circulation. Around the cloaca, in sexually mature animals, sit a set of glands the function of which was a mystery until quite recently. A study published in Scientific Reports in 2016 by a Belgian research group identified them as the source of a class of courtship pheromones. Large glycosylated proteins, about 20 kilodaltons in size, belonging to a family called the sodefrin precursor-like factors. These proteins are released into the water during the species courtship display, in which the male performs a slow, undulating dance in front of the female, with his cloaca held open, releasing a chemical signal that the female responds to by following him and eventually picking up a packet of sperm, a structure called a spermatophore, that he deposits on the substrate. There is no copulation. The sperm is transferred through the water by way of the spermatophore and the female's cloaca, an arrangement common to most mole salamanders and unfamiliar enough to humans that it tends to require a moment of explanation. The whole sequence takes perhaps 20 minutes. A successful pairing produces somewhere between 200 and 1,500 eggs, which the female attaches in clusters to plant material or rocks, and which hatch about 2 weeks later into small free-swimming larvae that look essentially like tiny adult axolotls. They have always looked like that. They will continue to look like that for the rest of their lives. This is what the body is: a pressure sensing, electric sensing, chemical sensing, suction feeding, three-way breathing salamander with elaborate external gills, soft pedicellate teeth, a mid-sized appetite, a slow-burning metabolism, and a reproductive system that operates by chemistry and choreography rather than by direct contact. None of it, on its own, is unprecedented in the amphibian world. Other salamanders have lateral lines. Other salamanders have ampullary organs. Plenty of amphibians breathe through their skin. Suction feeding is widespread in aquatic vertebrates. The pheromonal courtship is shared with the broader mole salamander family. What is unusual about the axolotl is not that it has any one of these traits, but that it has all of them in combination, holds them throughout its adult life, and combines them with a feature that does not appear in this complete form in any of its close relatives: the capacity to rebuild its own body after injury to a degree that no other vertebrate of comparable complexity has been documented to match. That capacity is the reason laboratories are interested in it. The lateral line, the gills, the pheromones, the suction feeding, the three respiratory systems, all of this is what makes the animal a functional organism in its environment. It is the standard biological infrastructure of a small aquatic carnivore, well-tuned, elegantly adapted, and moderately interesting. The regenerative system, on the other hand, is not standard. It is the thing the animal does that nothing else does on this scale. And it is the reason the graduate student in Vienna lowered the lights an hour ago and wrote a few notes in her log and turned away from the tank to go home for the night. She is not studying the axolotl because it is cute, or because it is endangered, or because the Aztecs built a god out of it, although all of those things are true, and all of them are part of why she ended up in this field rather than another. She is studying it because of what its cells do when it loses a limb. The animal, alone now in the dark room, drifts a few millimeters to the left and resettles. The cooling unit cycles on. The water moves slightly. Somewhere along the sides of its body, neuromasts register the movement and report it inward. Somewhere on its head, ampullary organs continue their constant low background reading of the electrical state of the room, the current in the wall, the heartbeat of the graduate student walking down the corridor outside, the standing field of the building itself. The animal is, in its quiet way, paying attention. It is paying attention to a world that no human in the building can sense. It will continue to do so until morning. The canal is quiet at this hour. It is somewhere around 2 in the morning in late autumn of 2024 in the southern district of Mexico City known as Xochimilco, and the brightly painted tourist boats, the trajineras that during the day carry Mexican families and foreign visitors slowly along the waterways with mariachi bands and food vendors aboard, are tied up motionless against the embankments, dark and empty. The water is the color of strong coffee. The air carries a complicated smell that is partly mud, partly damp vegetation, partly the diesel of the city beyond the wetland edge, and partly something organic and slightly sour that comes from the canal itself. From a distance, say, from the highway overpass that crosses the wetland a few kilometers to the north, Mexico City's lights stretch out in every direction to the horizon. 22 million people are settling into the deepest part of the night. Down at the waterline, the city is barely audible. A car passes somewhere far away. A heron, invisible in the reeds, shifts its weight and is silent again. About 40 centimeters below the surface, in a slow current near the edge of a chinampa, a wild axolotl is hovering above silt. It is dark olive green, faintly speckled with gold along the spine, and unless you knew exactly where to look, you would not see it. The pet store axolotl that lives in glass tanks in Berlin and Tokyo, the soft pink one, gills a pale flush, eyes black or red depending on the genetic line, is the descendant of generations of laboratory breeding and would, in this canal, glow against the dark substrate like a beacon and be eaten within an hour. The animal here is the original, wild type. The color evolution actually produced before human selection began rewriting the species in tanks. It is roughly the same size as the laboratory animal in Vienna, about 20 centimeters, but everything about how it sits in the water is different. Its breathing is slower. Its gills are pulled tight against the skull. It is paying attention to the canal in the way Chapter 3 described an axolotl paying attention to a tank, except that the channels carrying information here are far more complicated and the consequences of misreading them are far higher. This particular animal is somewhere between 50 and 1,000 of its kind that are still alive in the wild. The exact number is not known, and that uncertainty is part of the story of what has happened to the species in the last quarter century. The most recent comprehensive population census conducted by the National Autonomous University of Mexico, the great public research institution known by its Spanish acronym UNAM, was completed in 2014. At that time, biologists working under the direction of Luis Zambrano, an ecologist who has spent more than 2 decades on this single species in this single wetland, found roughly 36 axolotls per square kilometer in Xochimilco. The previous comprehensive census in 2008 had found about 100 per square kilometer. The one before that, in 2003, had found 1,000. The first systematic count, conducted in 1998, had found 6,000. The trajectory across those 4 data points is brutal in the way that population collapse trajectories tend to be brutal, not gradual, not linear, not slowing as it approached the bottom. Between 1998 and 2014, the wild population of Ambystoma mexicanum in its only natural habitat declined by roughly 99%. A new census is currently underway, the first since 2014. It is being coordinated by a UNAM biologist named Vania Mendoza and uses a combination of traditional sampling, nets, careful canal-by-canal walks, visual surveys, and a more recent technique called environmental DNA, in which water samples are filtered and the genetic material left behind by passing animals is sequenced and identified. The eDNA approach is well suited to a species this rare because it can detect an axolotl that swam through a particular section of canal hours or even days earlier without anyone needing to lay eyes on the animal itself. As of late 2024, the work is ongoing. The preliminary indications are not encouraging. A recent UNAM modeling exercise, based on current trends in habitat quality, invasive species pressure and water chemistry, suggests that wild axolotls may disappear from the Xochimilco canals entirely by the end of 2025 if conditions do not change. The authors of that model are the same people running the census. They are not given to dramatic statements. The number is the number. To understand how a species native to one of the longest continuously inhabited urban watersheds in the Americas got into this situation, it helps to back up and look at the wetland itself. Xochimilco today is a small remnant, perhaps 2% of what it once was. The original lake system of the Valley of Mexico was a connected network of 5 shallow freshwater and brackish lakes: Texcoco, Xochimilco, Chalco, Zumpango, and Xaltocan, that filled the floor of the high closed basin between the volcanic ranges to the east and west of what is now the city. The Aztecs, arriving in the valley in the early 14th century, did not just live around these lakes, they engineered them, Over the course of 2 centuries, they built up an elaborate hydrological system of canals, dikes, sluice gates, and most famously the chinampas, long rectangular agricultural platforms constructed by stacking layers of lake-bottom mud, decomposing vegetation, and reed mats to a height just above the waterline, anchored at the corners by living willow trees whose roots stabilize the structure from below. Each chinampa was a small island of extraordinarily fertile soil, surrounded by canals that provided irrigation, drainage, transport, and, critically, habitat. The system was used for agriculture, for fish farming, for the hunting of waterfowl, and incidentally for the continued thriving of a great many native species that found in the canals between chinampas a kind of artificial wetland edge that suited them well. Among those species was the axolotl. The chinampa system worked for somewhere around 1500 years. It was, by any reasonable measure, one of the more successful examples of sustainable urban agriculture in the human record, supporting the dense population of Tenochtitlan, the Aztec capital, which sat on an island in the middle of Lake Texcoco, without exhausting the resources it depended on. The Spanish, arriving in 1519, did not understand it. They saw a swampy, unhealthy landscape that needed to be drained for proper European-style agriculture and Christian-style cities to take root. Over the following 4 centuries, in stages, they drained it. Lake Xaltocan went first, then Zumpango, then most of Texcoco. Lake Chalco, the southernmost of the 5, was finally drained completely in the 1970s as part of a flood control scheme. Xochimilco itself, partly because of the dense network of chinampas, held its hydrology together better than the open lakes, partly because UNESCO designated it a World Heritage Site in 1987, survived. But it is now, ecologically speaking, a single small wetland surrounded on every side by one of the largest cities on Earth. The water that fills it is not the water that used to fill it. The original springs that fed Xochimilco have largely been pumped dry to supply Mexico City's drinking water demand. The shortfall is made up today by water from a single treatment plant at Cerro de la Estrella, which discharges processed wastewater into the canal system. The treatment is generally adequate. After heavy rain, when the city's combined sewer system overflows, it is not. Sewage enters the canals. Bacterial counts spike. The animals that live in the canals, the axolotl, the native crayfish Cambarellus montezumae, the silverside fish, several endemic plants, have to absorb the consequences of every storm. Then there are the fish. In the 1970s and 1980s, in an effort to provide a cheap protein source for the working-class neighborhoods around the wetland, the Mexican government deliberately introduced 2 species into the canals, African tilapia, Oreochromis niloticus, and Asian common carp, Cyprinus carpio. Both species are tough, fast-breeding, omnivorous, and well adapted to disturbed waters. Both established themselves immediately. Both, it turned out, eat axolotl eggs. They also eat the small invertebrates that adult axolotls hunt, and they outcompete axolotl larvae for the same food sources. The introductions were not malicious. They were a genuine attempt to address food insecurity in a poor district of a vast city. They were also, in retrospect, the proximate cause of a population collapse that has not been arrested in the 4 decades since. A wild axolotl in Xochimilco today is, in essence, surviving in a significantly hotter, dirtier, more crowded, more chemically variable, and more predator-rich version of the environment its species evolved in. It has not had time, in evolutionary terms, to adapt to any of these changes. The lake that shaped its biology over 10 or 15 million years was cool, oxygen-rich, predator-poor, and populated by the species the axolotl had been hunting and being hunted alongside since long before humans arrived. The lake it lives in now is none of those things. The animal is still doing what it has always done, hovering motionless in the silt, feeling for pressure waves and electrical fields, striking at small invertebrates when they drift into range. But the system around it has been reorganized by forces it has no biological response to. The carp arrives, the eggs disappear, the water warms, the bacterial load rises after a storm. Somewhere along the chain, the population thins. The thinning has been going on for 40 years. There is an effort to stop it, and the effort is interesting in its own right. UNAM's Laboratorio de Restauración Ecológica, founded by Zambrano in 2004, has been working with the local chinamperos, the small-scale farmers who still tend chinampas in the traditional Aztec manner, to convert sections of the canal network into what the team calls refugios, protected stretches of water in which semi-permeable barriers exclude tilapia and carp while still allowing water flow, in which the surrounding chinampa is farmed without pesticides or chemical fertilizers, and in which the original water filtering function of the chinampa system is allowed to operate as it did for 15 centuries before industrial agriculture. There are now somewhere around 48 of these refuges, distributed across approximately 5 linear kilometers of canal. The water inside them, biologists have measured, is meaningfully clearer than the water outside. The bacterial counts are lower. The native invertebrate populations are higher. And axolotls released into the refuges, both wild individuals translocated from worse habitat and captive bred animals from UNAM's own breeding population, have, in a study published in PLOS One in early 2025, been shown to survive, hunt, gain weight, and in some cases form what the researchers cautiously described as social associations with particular other individuals. 2 of the captive-bred animals released into the chinampa refuges during the 2024 study were eaten by great egrets, a common Mexican wetland bird, within the first 3 weeks. The team is now working on what they call predator awareness training, a kind of pre-release conditioning protocol intended to teach captive-raised axolotls to recognize and flee from avian silhouettes overhead. Whether such training can be effective in an animal whose primary perceptual channels are mechanical, chemical, and electrical, and whose visual system is poor enough that it likely cannot resolve a flying egret in time to react, is an open question. The team is testing it. They will publish what they find. This is what conservation looks like in this particular case, in this particular wetland, in this particular year. It is not heroic. It is the very slow work of trying to hold a small piece of ground against a much larger set of pressures, with a small budget and a small team and the consistent support of UNAM's Biology Institute and a relatively small number of local farmers who have decided that keeping the chinampas going is worth doing for its own sake. The chinampas, by the way, predate the axolotl crisis by more than a millennium. They were not designed for axolotl conservation. They turn out to be very good at it incidentally, because the same conditions that make them productive agricultural systems, clean water, intact native vegetation, biological filtering, the absence of industrial chemical input, also happen to be the conditions a wild axolotl needs in order not to die. The Aztecs, again, got something right that the modern world is now trying, with considerable difficulty, to recover. Back at the canal, the animal in the silt has not moved. It is still doing what it was doing an hour ago. Somewhere on the next chinampa over, in the dark, a small invertebrate has displaced a few cubic millimeters of water. The axolotl's lateral line registers the displacement. The animal does not respond. The disturbance is too small, too far, not worth the metabolic cost of a strike, but the reading is taken. The information enters the animal and is processed by whatever processes information in a salamander brain, and the animal continues to wait. It is good at waiting. Its species has been doing this here, in some version of this canal, for somewhere on the order of 10 million years. It does not know that the species has, in the last 0.25% of that time, lost 99% of itself. It does not know about Mexico City overhead, or about the treatment plant, or about the carp, or about UNAM, or about the census that is currently looking for it with environmental DNA samples. It is just an animal in a canal, doing what it has always done, in a place that may not contain it for very much longer. Cut off an axolotl's leg. This is, as a matter of routine, what regenerative biologists do. A young adult animal, healthy, perhaps 8 or 9 months old, is anesthetized in a dilute solution of a buffered amphibian anesthetic placed on a clean dissecting surface, and one of its forelimbs is amputated cleanly with a sharp blade just above the elbow. The cut bleeds for a few seconds and then stops. The animal is returned to its tank, where it recovers from the anesthetic over the following hour, and within a day it is hunting and feeding normally. The wound at the amputation site, examined under a dissecting microscope 24 hours later, has closed, not scarred over. Closed, covered by a thin, transparent sheet of new skin called a wound epithelium, which the animal's keratinocytes have produced and migrated into place over the course of the previous several hours. There is no scab. There is no obvious sign that anything is missing yet, except that the limb is shorter than the limb on the other side. This is the beginning of what is, by some distance, the most thoroughly studied regenerative process in any vertebrate. The wound epithelium is the first oddity. In a mammal, a wound of comparable severity would form a clot, attract neutrophils, the aggressive frontline cells of the inflammatory response, and develop over the following days into scar tissue dominated by collagen fibers laid down in a disorganized cross-hatched pattern by fibroblasts under the constant signaling of a growth factor called transforming growth factor beta. The mammalian repair process is remarkably effective at sealing the body against infection and stabilizing the structural integrity of the wound, and it produces, as a byproduct, a permanent fibrous mark that will never carry hair, will never sweat, will never quite match the surrounding skin in color or flexibility. The mammalian system, in evolutionary terms, prioritizes survival over restoration. Close the wound, get back to functioning, accept the scar. The axolotl does something different. The wound epithelium that closes the amputation site is not a scab, but a thin, almost transparent layer of living cells that lacks the structural feature called a basement membrane, the dense layer of extracellular matrix that normally separates the epidermis from the underlying tissue. The absence of the basement membrane matters because it means signaling molecules can pass freely between the wound surface and the cells underneath, and the cells underneath are about to do something that mammalian cells will not. They are about to step backward in their developmental history. Over the 3 to 4 days following amputation, the cells in the tissue immediately below the wound, bone cells, cartilage cells, muscle cells, the Schwann cells that wrap around peripheral nerves, the fibroblasts of the connective tissue, begin to de-differentiate. They shed the markers of their adult identity. They reactivate genes they have not used since the embryonic period. They become, in cellular terms, less specialized, not pluripotent stem cells exactly. They retain some memory of where they came from, but progenitors, cells with their developmental options reopened. By around day 7, a small mound of these de-differentiated cells has accumulated at the amputation site, organized beneath the wound epithelium into a structure called the blastema. The word is from the Greek for sprout, and the blastema is, functionally, what makes axolotl regeneration possible. It is a small, heterogeneous pool of cells with the capacity to become anything the missing limb requires, organized in a structure that knows where it is and what it needs to build. How the blastema knows what it needs to build is one of the central questions in the field, and the answer is still emerging. Part of it lies in the fibroblasts. When a fibroblast in the dermis of an axolotl's elbow region dedifferentiates and joins the blastema, it carries with it, encoded somewhere in its gene expression in ways researchers are only beginning to map, a kind of address. Elbow region fibroblast. The address is positional information, the cell's memory of where it sat in the body before injury, and the blastema as a whole reads the collective address book of its constituent cells to figure out what it is supposed to regenerate. A blastema formed at the wrist will build a hand. A blastema formed at the shoulder will build an entire arm. The cells know. The signaling that organizes this knowing has been worked on for decades and is still not fully understood. A research group led by James Monaghan at Northeastern University in Boston has, in a series of papers culminating in 2024 and 2025, focused on a small molecule called retinoic acid, a derivative of vitamin A that has been known since the 1970s to influence limb regeneration, and that turns out, in axolotls, to function as a kind of positional coordinate. The Monaghan Group's recent work, supported by the US National Science Foundation, has shown that what matters is not how much retinoic acid is present, but how it is being broken down. The enzyme that degrades retinoic acid, present in different concentrations in different parts of the limb, produces a gradient of the molecule that the blastema cells appear to use to figure out their position along the proximal distal axis of the regenerating structure. Using the gene editing tool CRISPR to disable a gene called SHOX, which is involved in human height regulation and which the axolotl shares, Monaghan's team showed that the blastema still regenerates a limb but builds it at the wrong length, proximal segments shortened, distal segments displaced. The shape comes from the same genes that build a human limb. The difference is not the genetic toolkit. The difference is that the axolotl can switch the toolkit back on in adulthood. Humans cannot, or at least not yet, and not without help. While the blastema is organizing itself, the immune system is doing something subtle and important. In a mammalian wound, the dominant immune cell type at the injury site during the first several days is the neutrophil, short-lived, aggressive, designed for clearing bacteria and damaged tissue and producing a strongly pro-inflammatory environment. In an axolotl wound, neutrophils are present in much smaller numbers, and the dominant immune cell type, even within the first 24 hours, is the macrophage, the longer-lived, more versatile cell that mammals also use, but typically only later in the healing process. Even more striking, axolotl macrophages at the wound site are predominantly anti-inflammatory in their molecular signature. They are signaling resolution rather than escalation. A 2013 study by James Godwin and colleagues working in Australia showed that depleting macrophages in axolotls before limb amputation completely blocked regeneration. The wound closed, but the limb never came back, the tissue underneath instead converting to a fibrotic stump that looked, structurally, very much like a mammalian scar. The macrophages, in some way researchers are still working out, are necessary for the regenerative program to run at all. The presence of the right immune cells in the right balance is what permits the dedifferentiation of the underlying tissue. Without them, the wound heals the way a mammalian wound heals, through scar formation, and the regenerative machinery never engages. Once the blastema has formed and the immune environment is correct, the rebuilding begins in earnest. The dedifferentiated cells start dividing rapidly. A 2016 study from the Tanaka lab in Vienna measured the cell cycle time of the relevant progenitor cells at around 5 days during peak regeneration, compared with around 14 days in homeostatic adult tissue. The cells, in other words, are dividing roughly 3 times faster than they normally would. They are also redifferentiating as they divide, taking on the identities of bone, cartilage, muscle, nerve, blood vessel, and skin cells, organizing themselves into the proper tissue layers, and following a developmental program that resembles, in many of its details, the embryonic limb development program, except that it is happening in an animal that is sexually mature, fully grown, and has been an adult for years. Around day 21, a small paddle-shaped structure has emerged from the wound surface. By day 30, fingers, or in the case of an axolotl forelimb, the 4 digits the species carries, have begun to differentiate from the paddle. By day 40 to 50, depending on the size and age of the animal, the new limb is essentially complete. Bone, cartilage, joint, muscle, peripheral nerve, blood supply, dermis, epidermis, the fine pattern of skin pigmentation. Functional. The animal uses it. If you photographed the regenerated limb against the original, you would have to look closely to tell them apart, and even then you might not be sure. There is, sometimes, a slight asymmetry in digit pattern. There is, sometimes, a small shift in proportion. Mostly, there is just a limb. This entire process, wound closure, dedifferentiation, blastema formation, patterning, redifferentiation, tissue assembly, runs to completion in roughly the same span of time it takes a human bone fracture to form a callus. 40 days. The axolotl in those 40 days has done something the human body cannot do at any timescale. It has done it before on the same animal. An axolotl that has lost a limb and regenerated it can lose the same limb again, regenerate it again, and do this a documented 5 or more times across its lifespan. Each successive regeneration is, on average, slightly less precise than the last. A 2024 study from a Mexican research group led by Edith del Moral Morales, comparing 8-month-old juveniles with 8-year-old adults, found that the older animals' blastomeres formed more slowly, expressed regeneration-associated genes at lower levels, and in some cases failed to regenerate at all by the time the juveniles had completed the process. There are limits. Older axolotls regenerate less well. Very large defects regenerate less well. Wounds in some tissues, large portions of the brain for instance, or the full thickness of the heart wall, do not regenerate completely even in young animals. The axolotl regenerative program is the most powerful vertebrate regenerative program known, and it is also bounded in ways that the popular framing of the species, the salamander that regrows everything forever, perfectly, flattens into something less interesting and less true. What it does, within those bounds, is still unprecedented in any animal of comparable complexity. The axolotl can regenerate a complete forelimb or hind limb, with all skeletal, muscular, neural, and vascular structures intact. It can regenerate its tail, including the segments of spinal cord running through it, with full functional connectivity. It can regenerate the lower jaw, the gills, the lens and retina of the eye, sections of the heart, sections of the spinal cord proper, and substantial portions of brain tissue. The axolotl spinal cord, when severed, does not form the glial scar that blocks axon regrowth in mammalian spinal cord injuries. The axons simply find each other again across the gap and reconnect. People who have been working on human spinal cord injury for their entire careers, and there are not many vertebrate biology problems harder than human spinal cord injury, look at axolotl spinal cord regeneration with a kind of professional astonishment that doesn't quite leave them no matter how many times they observe it. There is one more thing worth noting. While all of this is happening, While the cells are dedifferentiating and the blastema is organizing and the macrophages are signaling resolution and the new tissue is assembling. The axolotl behaves externally as if nothing unusual is going on. It eats, it hunts, it rests, it interacts with other axolotls in its tank in the normal way. Its metabolism does not radically increase. It does not, as far as anyone can tell, experience the regenerative process as a special state. The rebuilding of an entire limb with all of its cellular complexity is happening in the background of an otherwise ordinary salamander life. A graduate student feeding the animal a worm on day 18, watching the small paddle of new tissue at the wound site, sees an axolotl doing what axolotls do. The animal, presumably, sees nothing at all. It just continues. How any of this is possible, how a mature vertebrate can hold open the developmental programs that nearly every other tetrapod closes after embryogenesis, how its cells retain the capacity to step backward, how its immune system runs in a register that mammalian immune systems lost somewhere on the evolutionary path between amphibians and us, turns out to be tied in ways the laboratory work of the last 15 years has begun to make clear, to the single biological feature that has defined the axolotl since before there were laboratories or microscopes, or even a Spanish word for the animal. It does not metamorphose. It does not become an adult in the way most amphibians become adults, and whatever choice, whatever evolutionary process produced that one peculiarity in this one species, it carried with it a set of consequences for what the animal's body is capable of that is still, 160 years after Auguste Dumeril first noticed it, surprising the people who study it. In the spring of 1865, in the converted monkey house at the Jardin des Plantes, Auguste Dumeril noticed something he had not been expecting, and that, on first encounter, must have struck him as a kind of taxonomic prank. A small number of his axolotls, perhaps 3 or 4 out of a colony that by then numbered several dozen, had begun quite spontaneously to do the thing the species was supposed to be incapable of doing. Their gills had begun to recede, the dorsal fin running along their backs was shrinking, their skin was thickening, taking on a duller and grainier quality. Their eyes, previously lidless and slightly bulbous, were developing eyelids. Within a few weeks, the affected animals had stopped breathing through their gills entirely, were taking gulps of air at the surface of the tank, and bore, in their general body plan, an unmistakable resemblance to the terrestrial tiger salamanders that lived in ponds across Mexico and the southern United States. The animals had metamorphosed, they had grown up. Dumeril wrote it up. He was, by this point in his career, in the habit of writing up everything that happened in the colony, because the colony kept producing observations that no one had been expecting. The metamorphosed animals lost their distinctive larval features. They could no longer survive permanently underwater. Several of them died, apparently because the tank conditions Dumeril maintained, designed for aquatic adult axolotls, were no longer suitable. A few survived as small terrestrial salamanders for some time afterward, looking to a casual observer like a different species entirely, which in functional terms they essentially were. The same animal, the same genome, expressing a completely different body. The species' refusal to metamorphose, it turned out, was not absolute. It was a kind of standing inhibition, a developmental program held in check by some internal regulator, capable of being released under the right conditions and producing, when released, exactly the adult form that Cuvier had been looking for 60 years earlier and had been unable to find. The reason Cuvier had been unable to find it was not that the form did not exist. It was that, in the wild, almost no axolotl ever reaches it. To understand why, it helps to step back and look at the broader family the axolotl belongs to. Mole salamanders, the genus Ambystoma, comprise about 33 species distributed across North America from southern Canada down through the central highlands of Mexico. The closest relatives of the axolotl are the various tiger salamanders and the Mexican mountain salamanders that share the trans-Mexican volcanic belt. All of them, like most amphibians, share a basic developmental pattern. The egg hatches into an aquatic larva with external gills. The larva grows and feeds and accumulates mass, and at some point, triggered by a hormonal signal that begins in the brain and propagates outward through the body, the larva undergoes metamorphosis. Gills are reabsorbed, lungs take over, the body remodels itself for life on land. The animal climbs out of the water, becomes a terrestrial adult, and returns to water only briefly each year to breed. This pattern works well in habitats where the larval pond is unreliable, where it dries up seasonally or freezes or otherwise becomes uninhabitable for long enough that staying in it is more dangerous than leaving. In those environments, metamorphosis is the path through which the species persists. The salamander that fails to metamorphose in a pond that disappears every August simply does not survive to reproduce. Selection prunes the laggards. The genetic variants that allow flexibility, that delay metamorphosis when the pond is good and accelerate it when the pond is failing, become the dominant ones. The whole life cycle is calibrated to a world in which the water is temporary and the land is, eventually, where you have to live. Xochimilco was not that kind of world. The lake system that the axolotl evolved in, the connected series of high-altitude lakes in the Valley of Mexico, fed by springs and sustained by the volcanic geology of the basin, was exceptionally stable. It did not dry up. It did not freeze. It held a relatively constant temperature in the range of 15 to 17 degrees Celsius year-round. Food was abundant. Land predators, snakes, raccoons, certain birds were a serious problem for any salamander that left the water, while aquatic predators were comparatively few. Under those conditions, the calculation a metamorphosing amphibian normally faces, stay and starve or leave and possibly survive, was inverted. Staying was the better strategy. Leaving was the dangerous one. Selection began to favor the variants that delayed metamorphosis longer and longer, and at some point in the species' evolutionary history, somewhere between 10 and 15 million years ago by current estimates, it favored the variants that delayed metamorphosis indefinitely. The animals that never left the water were the ones that successfully reproduced. The animals that did leave were eaten. The trait, in technical biological language, is called pedomorphosis, or more loosely, neoteny, the retention of larval features into sexual maturity. Several other salamander species in the same Mexican volcanic region show various degrees of it. The axolotl is the most extreme case, an obligate pedomorph that under normal conditions never metamorphoses at all. The mechanism that holds the standing inhibition in place turns out to be, perhaps not surprisingly, hormonal. In a metamorphosing amphibian, the trigger for the whole transformation is the thyroid gland. The brain, specifically the hypothalamus, sends a signal to the pituitary. The pituitary sends a signal to the thyroid, and the thyroid responds by producing two hormones called thyroxine and triiodothyronine, abbreviated T4 and T3, which then circulate through the body and bind to thyroid hormone receptors in tissue after tissue, activating the latent developmental programs that drive metamorphosis. Gill resorption, lung development, skin remodeling. The whole transformation runs on this hormonal signal cascade, and in most amphibians, the cascade fires reliably as the animal approaches the right size and the right time of year. In the axolotl, the cascade is broken, but not in the place you might expect. The thyroid gland itself works. the thyroid hormone receptors in the tissues work. If you take an axolotl and inject it with thyroid hormone directly, bypassing the brain and the pituitary, the animal will metamorphose. Within 2 to 3 weeks of treatment, the gills begin to recede. Within 2 months, the transformation is complete. The animal becomes a terrestrial adult salamander, structurally indistinguishable from a tiger salamander of comparable size. The genetic toolkit for metamorphosis is intact. The cellular machinery for executing it is intact. What is missing is the upstream signal. The axolotl's hypothalamus does not, under normal conditions, tell the pituitary to tell the thyroid to start producing the metamorphic dose. The signal is suppressed at the brain level, and the result is a body that has, throughout its entire life, all the necessary equipment for becoming an adult salamander, but no instruction to do so. This explains Dumeril's observation in 1865. The handful of animals in his colony that spontaneously metamorphosed were, presumably, individuals in which the suppression failed, perhaps for environmental reasons, perhaps for genetic ones, and the cascade fired. They became what their cellular machinery had always been capable of becoming. Most of his colony, like most wild axolotls, never received the signal at all and stayed in the form their species had selected for. There is a question that follows from this, and the question is the part that begins to feel strange. If the axolotl carries the full machinery for metamorphosis, including functional thyroid receptors and an intact developmental program, and if this machinery can be activated by hormonal manipulation, then it is reasonable to ask what happens to the animal when the manipulation is performed. Not just in terms of body shape, the gill loss and the lung development and the skin thickening, but in terms of everything else the animal can do. What happens, it turns out, is that a great deal of what makes the axolotl interesting goes away. A metamorphosed axolotl can no longer regenerate limbs as well as a non-metamorphosed one. A 2014 study by James Monaghan's group, before he moved his attention to retinoic acid, compared limb regeneration in axolotls before and after thyroid hormone-induced metamorphosis. The metamorphosed animals regenerated more slowly, by a factor of roughly 2, and produced limbs with a higher rate of structural abnormalities, particularly in the digits. A separate Turkish research group led by Turan Demirkan found, in a study published the same year, that several tissue regeneration capacities were reduced or lost entirely after metamorphosis, with corresponding shifts in the animal's microbiota and gene expression. The cellular plasticity that the axolotl carries through its entire life, the dedifferentiation, the blastema formation, the reactivated embryonic programs, diminishes when the animal completes the metamorphic transition. The wound healing tilts toward the mammalian pattern. Scar tissue begins to form. The glial scar, which the axolotl spinal cord normally avoids, becomes more likely. The macrophage profile shifts. The tissue, in short, stops behaving like axolotl tissue and starts behaving like the tissue of an ordinary adult salamander, which is to say, not particularly remarkable. This is the hinge of the whole story, and it is worth stating clearly before going further into it. The axolotl's regenerative capacity is not separate from its refusal to metamorphose. The two appear increasingly to be aspects of the same underlying biological condition. The animal that does not grow up retains, into adulthood, the cellular flexibility that all vertebrate embryos have during development. The animal that grows up loses it, in the same way that other vertebrates lose it when they grow up, in the same way humans lose it before we are born. Maturity, in this lineage, is a kind of closing. Adulthood is the moment the developmental options are sealed. The axolotl, by never quite reaching adulthood, never quite seals them. The Aztecs, watching the animal in their canals 900 years ago and building a god out of it, named it after Xochimilco, the deity who refused his sacrifice, who fled into the water rather than complete the transformation expected of him, who chose to become this strange, small creature rather than die properly as a god. The myth, as a piece of biology, is uncomfortably close to accurate. The animal really did refuse a transformation. It really did remain, in the developmental sense, a child. And what that refusal granted it, in return for the costs of staying in the water and never seeing dry land, is the ability to do something with its own body that no fully adult vertebrate of comparable complexity has been observed to do. Dumeril in 1865 did not yet know any of this. He saw a few of his animals turn into terrestrial salamanders and wrote it up. He did not connect the metamorphosis to the regenerative observations he would publish 2 years later. The connection took another century and a half to clarify and is still being worked out in laboratories from Vienna to Boston to Mexico City. But the pattern was already there in the colony in the spring of 1865 in the converted monkey house. Some of the animals grew up and lost the gift. Most of them stayed children and kept it. And the gift, the strange, partial, bounded, biologically remarkable gift of a body that can rebuild itself, turned out to be inseparable from the staying. There is a particular kind of laboratory experiment that, when you describe it in plain language, sounds almost cruel, and which, when you understand what it reveals, turns into one of the more philosophically loaded pieces of biology produced in the last 20 years. You take 2 axolotls of approximately the same age, the same size, the same genetic background, siblings from the same clutch, ideally, raised in adjacent tanks under identical conditions. You leave one alone. The other you treat with thyroxine, the metamorphic hormone the animal's brain normally declines to release, at a controlled dose, over a period of 2 to 3 weeks. The treated animal undergoes the transformation that wild axolotls almost never undergo. Its gills recede and disappear, its dorsal fin shrinks and is reabsorbed, its skin thickens and changes texture, its eyes develop functional eyelids. Within roughly 2 months, it has become, externally and internally, a terrestrial adult salamander, a more or less ordinary-looking ambystoma of the kind that exists in many habitats across North America and produces no scientific astonishment at all. You then take both animals, the original neotenic one and its metamorphosed sibling, and amputate a forelimb from each. You return them to their tanks. You wait. The first animal regenerates its limb on the schedule, and with the precision described earlier in this script, wound epithelium within hours, blastoma by day 7, paddle-shaped early limb by day 21, fully formed and functional limb by day 45 or so. The second animal does not. Its wound closes more slowly. The blastema, when it forms at all, forms with a delay and at reduced cell density. The cell cycle acceleration that drives normal regeneration is muted. The dedifferentiation of underlying tissue is partial. The macrophage profile at the wound site shifts away from the resolution-favoring pattern and towards something closer to the mammalian inflammatory pattern. The new limb, when it eventually emerges, is malformed. The digits are abnormal, sometimes too few, sometimes fused, sometimes structurally disorganized in ways that compromise function. Regeneration takes roughly twice as long. The limb that results is, in the technical assessment of the researchers running the experiment, a partial failure. The animal has not been damaged, it has not been poisoned, it has not been deprived of any nutrient or environmental requirement. It has simply been allowed, caused more accurately, to do what its species spent 15 million years declining to do. It has grown up, and in the process of growing up, it has lost almost everything that made it biologically remarkable. This experiment, in various forms, has been repeated by laboratories around the world since the early 2000s. James Monaghan's group at Northeastern University published a careful version of it in 2014 in the journal Regeneration. The Turkish researcher Turan Demirkan and colleagues published a parallel set of findings the same year. More recent work, including a 2023 study from the Mexican neuroendocrinologist Aurea Orozco's lab at the National Autonomous University of Mexico, has begun to map the molecular details of what the metamorphic transition does to the regenerative program at the level of gene expression and tissue microenvironment. The picture that has emerged across all of these studies is consistent and, in its implications, unusual. The axolotl's regenerative capacity is not a separate bonus feature bolted onto an otherwise normal salamander. It is structurally entangled with the species' refusal to mature. The flexibility that lets cells dedifferentiate, the immune environment that favors resolution over scarring, the cell cycle dynamics that drive blastema growth, the absence of the glial scar in spinal injury, the wound epithelium chemistry, all of it appears to depend on the animal remaining in the developmental state it occupies for life under normal conditions. When the metamorphic signal fires and the body completes its transition to adulthood, that state collapses, and with it, most of what makes the axolotl interesting to a regenerative biologist. Maturity in this lineage is a kind of biological closing of doors. The doors do not, once closed, easily reopen. This is the part of the science that is worth pausing on, because it carries an implication that is genuinely strange. The axolotl, alone among well-studied vertebrate models, is essentially a fully grown animal that retains the cellular flexibility of an embryo. Every other vertebrate biology has been able to examine in detail, including ours, closes that flexibility during development and never reopens it. Embryonic cells in a mammalian fetus can do many of the things axolotl cells can do. Adult mammalian cells cannot. Somewhere between conception and birth in our lineage, a set of developmental options is sealed. Somewhere between hatching and the first year of life, in most amphibians, the same sealing happens. The axolotl simply does not do this. It carries the open developmental state forward, indefinitely, into a body that grows, ages, and reproduces as if it were an adult while retaining, at the cellular level, the responsiveness of something that is still, in some important sense, in the process of being made. What the metamorphosis experiments demonstrate is that this is not an accident of timing, or a leftover trait the species happens to retain. It is the active condition under which the regenerative capacity exists. Force the animal to complete its development and the capacity goes. Permit the animal to stay where it is and the capacity stays. The choice, if such a word fits, which it does not quite, since the animal is not making a choice, only inheriting one, is between growing up and being able to rebuild yourself. The two are, in this lineage, mutually exclusive. The axolotl took the second option. Every other vertebrate, including us, took the first. At which point it becomes difficult, even for someone trying very hard to stay within the bounds of careful biological description, not to notice that the Aztec mythology recorded by Bernardino de Sahagún in the 16th century, the story of the god Xochimilco, who refused to be sacrificed, who fled into the water rather than complete the transformation expected of him, who chose to become a small, strange creature rather than die properly as a god, described the biological situation of this animal with an accuracy that should not, by any reasonable accounting, have been available to a culture without microscopes or hormonal endocrinology. There are several ways to interpret this, and I want to be careful about which one I land on. One interpretation is that it is coincidence, and that human cultures generate so many stories about so many animals that some of them, by sheer probability, will turn out to map onto the underlying biology in retrospect. This is, statistically, not implausible. Another interpretation is that the Aztec naturalists who watched these animals for centuries saw something the European biologists did not, that the animal, in its persistent juvenile form, presented as something that had refused a transformation other salamanders completed, and that the mythology encoded an observation rather than inventing one. This is, anthropologically, also not implausible. A third interpretation is somewhere in between, that the myth came first, that the animal happened to fit it, and that the fit is real, but the causality runs in the wrong direction for it to count as ancient knowledge of biology. I am genuinely not sure which of these is correct. What I am sure of is that the alignment is unusually clean. The animal in the canal is, in a literal biological sense, the creature in the myth. The refusal, the flight into the water, the staying in the strange form rather than completing the cycle, all of it maps. And whether that mapping is a coincidence or a piece of careful pre-scientific observation or something stranger than either, the fact of the alignment is not in dispute. Here, in the deepest part of this script, is where the discovery principle the channel is built around, the proposition that every animal is more than you think, does its hardest work. The axolotl is more than you think because it is a salamander that does not grow up. It is more than you think because not growing up is the price of admission for what its body can do. It is more than you think because the people who lived alongside it for 1500 years before any European had heard of it built a god out of it whose mythology happens to describe, with disconcerting precision, the biological fact that its species refused a transformation and remained for evolutionary reasons that probably have to do with the stability of a lake system that no longer exists in the form its ancestors moved through. The animal in the canal has been a child for 10 million years. The Aztecs noticed. The Europeans, working with better instruments and a worse mythology, took another 400 to catch up. A note about what this means for human biology before continuing, because it is the question almost everyone asks at this point, and because the honest answer is more interesting than the popular answer. The popular framing, which has appeared in headlines for at least 2 decades, is that axolotl research is on the verge of enabling human limb regeneration. This is not accurate. The careful framing, which the actual researchers in the field are consistent in offering, is that axolotl research is helping us understand the cellular and immunological conditions under which complex tissue regeneration becomes possible, and that the near-term clinical applications over the coming decade or two are likely to be in scarless wound healing, in fibrosis prevention, in spinal cord repair, and in cartilage and joint regeneration, rather than in regrowing entire limbs. The US National Science Foundation, in a 2025 statement summarizing the Monaghan Lab's recent work, put it about as plainly as a federal funding agency ever puts anything. We are still a long way from humans regrowing limbs. The phrase "long way" is doing real work in that sentence. The questions being answered now are foundational. The applications, when they arrive, will arrive in narrower domains than the popular framing suggests, and on timescales measured in decades rather than years. What the axolotl is teaching us is not that the human body can be made to regrow itself. What it is teaching us is that the genetic toolkit for tissue regeneration is broadly conserved across vertebrates. Most of the genes the axolotl uses to rebuild its limbs, humans also have, and that the difference between an animal that can rebuild and an animal that cannot is not the genes themselves, but the regulatory state in which the genes sit. The axolotl keeps its regulatory state open. We close ours during development and never reopen it. The question of whether we could be persuaded, chemically, genetically, immunologically, to reopen specific aspects of that state in specific tissues under specific medical conditions is the question the field is actually working on. The answer, when it comes, will be partial. It will not look like an axolotl. It will look like better wound healing, fewer scars, faster nerve recovery, more complete cartilage repair, and possibly eventually modest reconstructive capacity in tissues currently considered beyond reach. Whether that arrives in 20 years or 50 or 80 is, at this point, a matter of how hard the problem turns out to be once the next layer of mechanism is mapped. The axolotl is the model system telling us where the next layer is. The animal itself, of course, has no idea any of this is going on. It has been hovering above silt for 10 million years, eating worms when worms drift past, shedding limbs in territorial disputes and growing them back, refusing to grow up because the lake it evolved in did not require it to. The Aztecs watched it and made it a god. The Europeans collected it and got it wrong. The laboratories are now spending billions trying to understand what its cells are actually doing when no one is paying attention. It does not know any of this. It just continues. The lake is dark, the water is cool, the gills are barely moving. Somewhere overhead, a great egret turns its head, and the animal, registering nothing, drifts a few millimeters to the left and resettles. If you printed the complete genome of an axolotl, every nucleotide of every chromosome in plain text at standard typeface and standard line spacing, the resulting document would run to roughly 3,200,000 pages. Bound as books, it would fill a substantial public library. Stacked as paper, it would reach the height of a 6-story building. The same biological information in a single human cell occupies about 320,000 pages by the same standards. The axolotl genome is, by total length, 10 times larger than ours. This was not known until 2018, and the reason it took so long to find out is that, until quite recently, no one could read it. Genome sequencing as a routine technology dates from the early 2000s. The Human Genome Project, completed in 2003, produced a draft of the 3 billion base pair human genome at a cost of several $billion, and the work of thousands of scientists across more than a decade. By the early 2000s, technological improvements had brought the cost of sequencing a vertebrate genome down by several orders of magnitude, and a long list of model organisms, mouse, zebrafish, chicken, various primates, had been read end to end. The axolotl, despite being one of the most important developmental and regenerative biology models in the world, was not on that list. It had not been read because it could not be read. The standard sequencing tools of the period worked by chopping a genome into millions of short fragments, sequencing each fragment and computationally reassembling the pieces by looking for overlapping sequence, a method that depends critically on the assumption that the original genome contains enough unique sequence for the overlap-finding software to figure out which fragment belongs where. The axolotl genome violated that assumption catastrophically. Most of its 32 billion base pairs are made up of long, repetitive sequences, a class of mobile genetic elements called long terminal repeat retrotransposons, which have copied and pasted themselves throughout the genome over evolutionary time and which look, to a sequence assembly algorithm, essentially identical to one another. The fragments could be sequenced, they could not be reassembled. The genome remained, in any practically usable form, a black box. The breakthrough came from an international team led by Eli Tanaka at the Institute of Molecular Pathology in Vienna, Siegfried Schleusnig at the Heidelberg Institute for Theoretical Studies, and Gene Myers, the bioinformatics pioneer who had played a central role in assembling the first human genome 2 decades earlier. Working with a long-read sequencing technology developed by Pacific Biosciences, a method capable of reading much longer fragments than earlier tools, long enough to span individual repetitive regions and resolve them. And with new assembly software the team built specifically for the problem, they generated more than 72 million long sequence reads from a laboratory axolotl and computationally stitched them into the most complete vertebrate genome ever assembled at that scale. The result was published in Nature in early 2018. A follow-up paper in PNAS in 2021 organized the sequence into the animal's full set of 14 chromosomes. The axolotl genome, after a century and a half of being a model organism that no one could read, was finally legible. What it revealed turned out to be both more and less than the field had been hoping for. Less in the sense that the genetic toolkit underlying axolotl regeneration is, in its broad outlines, the same toolkit that mammals carry. Most of the genes the axolotl uses to build a limb during development and to rebuild one after amputation have direct human counterparts. The dedifferentiation programs, the patterning genes, the immune signaling cascades, the cell cycle regulators, almost all of it has been catalogued before in mice and chickens and fruit flies and humans, in different combinations and with different regulatory contexts. The axolotl is not regenerating limbs because it has secret genes no one else has. It is regenerating limbs because it can switch on in adulthood a set of genes that other vertebrates can only switch on during embryonic development. The genes are conserved. The control is what differs, more in 2 specific and slightly puzzling ways. The first is that one of the standard developmental genes, PAX3, which in mammals plays an essential role in muscle and neural development and in the migration of neural crest cells during embryogenesis, is, in the axolotl, simply absent, not silenced, not modified, not present in some unusual form. It is missing from the genome. A close relative of the gene, PAX7, has taken over its functions, performing both its own normal role and the role PAX3 plays in mammals. This is the kind of finding that, when it appears in a Nature paper, sends a small shockwave through developmental biology, because PAX3 had been considered one of the unambiguously conserved master regulators of vertebrate development. To find an entire functional vertebrate organism that has dispensed with it, that runs its developmental program through a single gene where mammals run it through 2, is the kind of discovery that opens questions rather than closing them. How long has the axolotl been without PAX3? Did its ancestors have it and lose it? Or did the lineage that became the axolotl branch off before the gene duplication event that produced the mammalian arrangement? What does the absence of PAX3 tell us about the redundancy and flexibility of developmental regulation more broadly? These are questions the field is still working on. The simple fact that the gene is gone and the animal is fine is the part that is established. The second puzzle is the size of the genome itself. 32 billion base pairs is not just a large vertebrate genome. It is, when the Tanaka Group's work was published in 2018, the largest vertebrate genome that had ever been completely sequenced. It is also not, as far as anyone has been able to determine, doing anything obvious that smaller genomes do not do. The axolotl is not a large animal. It is not a particularly long-lived animal, a lifespan of 15 to 25 years in captivity, less in the wild, in a range comparable to many salamanders with much smaller genomes. It is not unusually complex in its anatomy, its behavior, or its physiology. By every measure that genome size has historically been thought to correlate with, the axolotl should have a genome of 1 or 2 billion base pairs, in line with most amphibians of comparable build. Instead, it has 32. Most of that extra length is non-coding. The introns and intergenic regions are dramatically expanded relative to mammals, and the expansion is largely accounted for by accumulated retrotransposon insertions over deep evolutionary time. Whether this expansion is functionally important, or whether it is simply genomic real estate the species has tolerated because nothing has selected against it, is genuinely unclear. There are arguments on both sides. There are some indications that very long introns in certain developmental genes correlate with the slow timing of axolotl development, that the sheer length of these genes affects how quickly they can be transcribed, and that the slow transcription rate may interact in some way with the regenerative program. There are other indications that the bulk of the expansion is, biologically speaking, surplus. The honest summary is that we have a complete sequence and a very partial understanding of what most of it does. Some smaller findings from the same body of work are worth noting. The axolotl carries a small number of genes that appear to be amphibian-specific or even axolotl-specific, genes without obvious counterparts in mammals, and a subset of these are expressed strongly in regenerating limb tissue. They are candidates for the species-specific component of the regenerative machinery, although whether they are essential or merely correlated with the process is still being worked out. The genome also contains an unusual pattern of repeat distribution that suggests a long history of relatively low recombination. The sort of pattern you would expect in a species with limited gene flow between populations, which fits with the axolotl's restricted historical range in a single Mexican lake system. What the genome does not contain, despite extensive looking, is anything that would amount to a clean explanation of the animal's regenerative capacity. There is no master regeneration switch. There is no single gene that, when transferred to a mouse or a human cell, would confer the ability to rebuild a limb. The capacity is distributed across hundreds of genes, across regulatory contexts, across the cellular and immunological state the animal carries throughout its life. And the genome is necessary but not sufficient to understand it. The genome tells you what the toolkit contains. It does not tell you how the toolkit is being used, or why this particular animal can use it in adulthood when others cannot. This is where the field has settled. The genome is sequenced, the mechanisms are partially understood. The connection to neoteny is increasingly clear. The therapeutic implications are being worked out in narrow, careful experiments rather than broad, sweeping ones. And the central biological mystery, how an animal with the same basic genetic toolkit as a mouse manages to do in adulthood what mice can only do as embryos, has been refined from a question with no answer into a question with several partial answers, none of which yet fits together into a complete picture. The animal at the bottom of its tank, of course, is doing none of the work involved in making any of this make sense. It is hovering above the gravel, its gills are pulled tight against the skull. The 32 billion base pairs encoded in each of its cells are, at this moment, being transcribed and translated into the proteins that maintain the animal's quiet, slow, attentive existence. The breathing, the circulation, the sensory monitoring, the digestion, the constant low metabolic hum of being a living creature. The genome is doing its work. The animal does not need to know how. There is a question that follows directly from everything described so far, and oncologists who hear about axolotl regeneration for the first time tend to arrive at it within about 30 seconds. If this animal's adult cells routinely step backward in their developmental history, de-differentiating into something resembling embryonic progenitors, dividing rapidly, reorganizing themselves into new tissue, and doing all of this on a regular basis throughout the animal's lifetime, then why, by every reasonable expectation of how cancer works, is the axolotl not riddled with tumors? Cancer in mammalian biology is fundamentally a disease of cellular misbehavior. Cells that should remain in a stable, differentiated state begin dividing when they should not, ignore the signals that ought to stop them, evade the immune surveillance that should clear them, and accumulate further mutations as they go. The conditions a tumor requires: uncontrolled proliferation, escape from differentiation, resistance to programmed cell death, the ability to remodel surrounding tissue, are conditions that read, on paper, almost like a list of features the axolotl deploys deliberately every time it regenerates a limb. The animal does, at the cellular level, many of the things a developing tumor does. It just does them in a coordinated, regulated, reversible way, and at the end of the process, there is a functional limb rather than a malignant mass. What it almost never does is develop actual cancer. This is not, it turns out, simply an absence of observation. Researchers have looked carefully. Going back to experiments in the 1960s and 1970s, axolotls have been exposed to potent chemical carcinogens, substances that reliably produce tumors in rodents at standard doses, and their tumor rates have been recorded as strikingly low. Spontaneous tumors in laboratory axolotl colonies are rare. Induced tumors, when they appear at all, frequently fail to progress in the way mammalian tumors progress. They stay localized, regress, or are simply contained by the animal's biology in ways that mammalian biology cannot match. The species is not perfectly cancer-resistant. No animal is, but it sits at the high end of the vertebrate cancer resistance spectrum, in the same general territory as elephants and bowhead whales, both of which have evolved their own sophisticated tumor suppression strategies. The axolotl's strategy looks, from what is currently known, to be different from either of those. A central piece of the picture involves a gene called TP53, which produces a protein called P53 that is sometimes described in slightly grandiose textbook language as the guardian of the genome. The function is real, even if the language is overheated. When a mammalian cell sustains DNA damage from radiation, from a chemical insult, from an error during replication, P53 is what notices. It halts the cell cycle to allow repair. If repair is not possible, it triggers the cell's self-destruction. The protein is, in a meaningful sense, the cell's quality control inspector, and mutations in TP53 are found in roughly half of all human cancers. When the inspector fails, damaged cells accumulate, divide, and eventually do what damaged cells do. What makes the axolotl version of P53 unusual is not that it is missing or overactive. It is the specific structure of the protein. Sequence comparison work, most extensively a 2007 study published in BMC Ecology and Evolution by a Canadian group led by Stéphane Roy at the Université de Montréal, found that the axolotl P53 carries several amino acid substitutions at positions that, in the human protein, are associated with reduced tumor suppressor activity. Specifically, residues at positions 155 and 157, which in human cancers are sometimes mutated to forms that compromise the protein's function, sit in the axolotl protein in the cancer-associated configuration as the standard arrangement. And the axolotl protein in that configuration works perfectly well. It still detects DNA damage. It still halts cell cycle progression when needed. It still triggers programmed cell death in irreparably damaged cells. The amino acid changes that would cripple a human tumor suppressor are, in the axolotl, just how the gene normally reads. The most plausible interpretation is that some of these substitutions evolved to maintain protein stability at the cool body temperatures the axolotl operates at, around 15 to 17 degrees Celsius, well below mammalian core temperature, and that the configuration that works for an animal at 15 degrees would have been disastrous in a mammal at 37. The cancer resistance, however, does not appear to rest on p53 alone. The Roy Group's experiments demonstrated something further and more interesting. When they used a small molecule called pifithrin alpha to block p53 signaling in axolotls, two things happened simultaneously. The animal's regenerative capacity collapsed, limb amputations failed to regenerate normally, and the cellular markers of intact tumor suppression were lost. The same molecule that broke regeneration also broke cancer resistance. The implication is that, in this species, the two capacities are not independent properties that happen to coexist in the same animal. They are, at some level, two outputs of a single underlying regulatory system. The same machinery that lets an axolotl rebuild a limb without producing a tumor is what keeps it from producing tumors under more ordinary conditions. Tight cellular regulation, careful coordination of proliferation and differentiation, an immune environment that resolves rather than escalates. The whole package serves both the regenerative function and the cancer suppressive function at once. There is more to it than just p53. Axolotls appear to maintain unusually robust DNA repair mechanisms, the cellular machinery that fixes damage before it can be propagated through cell division. Their cell cycle checkpoints, which monitor the integrity of the genome at each stage of replication, are tight. The reduced inflammatory tone of their tissues, the same tone that lets regeneration proceed without scarring, also limits the chronic inflammation signal that, in mammals, contributes meaningfully to cancer initiation. Some recent work has also pointed to the role of cellular plasticity itself as a possible protective mechanism. Cells that retain the ability to dedifferentiate may, paradoxically, be less likely to transform because the regulatory networks holding them in flexible but controlled states are more robust than the networks holding terminally differentiated cells in place. The axolotl's cellular state is, in this view, controlled flexibility, open enough to permit regeneration, closed enough to prevent malignancy. The control is the trick. Mammalian cells trade flexibility for stability. Axolotl cells trade neither, and have evolved the regulatory architecture to manage both at once. Whether any of this can be translated into human cancer therapy is the question the popular framing inevitably reaches for, and the honest answer is that it is too early to say with confidence. The mechanisms are genuinely interesting. The conservation of the underlying genes between the axolotl and humans is real. But translating an integrated, evolved over millions of years regulatory system from one species into another is not the kind of thing that happens through a single intervention or a single drug. The clinical applications most likely to emerge from this line of work over the coming decade or two are probably in tumor types where the relevant regulatory pathways can be modulated pharmacologically in narrow, specific ways. The dream of an axolotl-derived universal cancer prevention is, at least in the framing the actual researchers offer, not on the near horizon. What is on the horizon is a clearer understanding of why some vertebrates manage cellular flexibility without paying the cancer cost, and how the architectural features that make that possible might be approximated partially and imperfectly in human tissue under medical supervision. The animal in the tank, of course, is not in the business of curing human cancer. It is in the business of being an axolotl, which involves, among other things, holding open a regulatory state most vertebrates have closed by adulthood, dividing cells in coordinated bursts when injury demands it, and avoiding, almost as a side effect, the cellular catastrophe that lies in wait for any organism whose cells are doing this much rearrangement. The avoidance is not magical. It is the product of a tightly coupled set of mechanisms that have been refined over evolutionary time to keep flexibility from becoming chaos. The axolotl is not immune to cancer because it has solved the problem. It is rarely cancerous because the same biology that lets it regenerate has, built into it, the regulatory discipline that keeps regeneration from going wrong. Somewhere in the dark of the laboratory, the animal continues. Its cells divide and do not divide. Its tissues remain in their proper configurations. The 32 billion base pairs are read, transcribed, translated, copied with high fidelity, and corrected when the copying fails. The system holds. It has been holding in this particular configuration for a very long time. In the autumn of 1962, a fisherman somewhere on Foot Lake, near the small town of Wilmar in west-central Minnesota, pulled a tiger salamander out of the water that did not look quite right. Tiger salamanders in that part of the upper Midwest are normally dark-bodied with yellow blotches, the kind of thick-set terrestrial amphibian that hunts in damp soil and breeds in shallow ponds in early spring. The animal in his hand was nearly white. Its skin had the translucent quality of an albino. Its eyes were red. It was, by the genetic accident that produces such individuals, lacking functional melanin production, which in a wild salamander is essentially a death sentence. Albino animals stand out against any natural substrate and rarely survive long enough to reproduce. This one had. The fisherman kept it for almost a year in a setup of his own devising before deciding he did not know quite what to do with it. He gave it to a biologist named Rufus Humphrey at Indiana University. Humphrey ran a laboratory axolotl colony, the American descendant by then several generations removed of the 5 animals that had been imported from Europe in 1935, and that, in turn, descended from Auguste Dumeril's Paris colony of 1864. He had been working with axolotls for decades. He was also one of the few people in the world who had thought carefully about the genetic relationship between axolotls and their nearest relatives in the tiger salamander complex, and he understood, in a way most amphibian biologists at the time did not, that the 2 animals were genetically close enough to interbreed under laboratory conditions if you were patient with the technique. He had been waiting more or less for an opportunity like this. He performed the cross. Using in vitro fertilization, he combined sperm from the albino tiger salamander with eggs from his own axolotl colony, and he raised the resulting hybrids. The hybrids were viable. They retained, generation after generation, the gill morphology and aquatic lifestyle of axolotls, the dominant trait when crossed with a metamorphosing tiger salamander. They also carried in recessive form the albino trait from the Minnesota animal. With selective breeding over the following years, Humphrey produced axolotls that were homozygous for the albino allele, animals that looked externally like axolotls in every way except that they were white, with red eyes and pale gold-tinted skin. These animals were beautiful, and for laboratory purposes they were also extremely useful. The lack of melanin made it dramatically easier to observe internal structures during early development, to track the migration of pigmented cells in transplant experiments, and to perform the kind of fluorescent imaging that has become standard in modern developmental biology. The albino axolotl became, almost overnight, the laboratory strain of choice for many kinds of research. It still is. The strain is called a /A in the genetic shorthand, and a substantial fraction of the laboratory axolotls used in regenerative biology research worldwide carry it. What the strain also is, technically, is a hybrid. Almost every albino axolotl now living, and through them, much of the global laboratory population and a significant portion of the pet trade, carries genetic material from a tiger salamander. The introgression is small, but it is real. A 2017 study by S. Randall Voss and colleagues at the University of Kentucky, using modern sequencing tools to compare the genomes of laboratory axolotls against wild-caught reference animals, identified specific tiger salamander DNA segments, including the region around the TIR gene, which encodes the tyrosinase enzyme responsible for melanin synthesis, distributed across the laboratory genome. The albino allele, the A/A, is itself a tiger salamander allele. It was never an axolotl mutation. It was carried into the species by Humphrey's deliberate cross, and from his Indiana colony it spread through generations of laboratory exchange into colonies around the world. The pet axolotl in a tank in Manchester or Berlin or Tokyo is, in the most precise genetic sense, not exactly the same animal as the dark salamander in the canals of Xochimilco. It is mostly that animal, with measurable contributions from a hybridization event 60 years ago in Indiana, layered on top of inheritance from a colony of 34 wild-caught Mexican animals shipped to Paris 160 years ago. The pet is a particular outcome of a particular human history. It has been bred, selected, recombined and transferred between continents and institutions for so long that asking whether it is truly the same species as its wild ancestors is no longer a clean question. The captive population, by every available estimate, numbers in the hundreds of thousands. There are laboratories in Vienna, Boston, Lexington, Mexico City, Beijing, Hanover and dozens of other cities maintaining substantial colonies for research. There are pet owners, many of them young, many of them introduced to the species through Minecraft, through TikTok, through a 50 peso note, through a school field trip to an aquarium, keeping axolotls in glass tanks across at least 100 countries. There are commercial breeders in Japan, in the United States, in the Czech Republic, in the United Kingdom, producing animals in industrial quantities for the pet trade. The species in captivity is so abundant that the Convention on International Trade in Endangered Species has had to develop specific protocols to distinguish legally bred captive animals from the few wild collected animals that occasionally enter the trade illegally. The wild population, meanwhile, sits at somewhere between 50 and 1,000 individuals. The arithmetic of this is worth letting sit for a moment, because it is one of the genuinely strange situations in modern conservation biology. The axolotl is not rare. It is, in absolute numerical terms, one of the more abundant amphibian species on Earth. There are more axolotls alive right now than there are individuals of most large mammal species, more than there are individuals of the great apes combined, more than there are individuals of many fish species considered commercially significant. They are everywhere. Tanks in classrooms, tanks in research institutes, tanks in living rooms, tanks in pet shops. The species is, in the captive sense, thriving. The wild population, in the place where the species evolved, is on the edge of disappearance. This is the captive wild paradox in its sharpest form, and it does not have a clean resolution. The captive animals are not, in any straightforward way, a substitute for the wild ones. The genetic narrowness of the laboratory population, the consequence of being descended from a small founder group with limited subsequent introductions and then deliberately inbred to fix particular traits like the albino allele, means that laboratory and pet axolotls have lost much of the genetic diversity that wild Xochimilco animals carry. Diseases that wild populations have evolved resistance to may sweep through captive populations with no defense. Behavioral traits, courtship preferences, foraging strategies, habitat selection have been shaped by tank life rather than by selection pressures in a complex wetland ecosystem. The hybrid contribution from Humphrey's 1962 cross, while small, may interact with the introduction of captive-bred animals into wild habitat in ways that researchers are only beginning to study. UNAM's conservation team has been clear that, while captive breeding plays an important role as an insurance population, the work of actually saving the species cannot be reduced to producing more axolotls in tanks. The work has to be saving the wetland the species evolved in. There is also a quieter aspect to all of this, which has to do with what the captive animal represents culturally. The pet axolotl is pink and smiling and small, and it has been embraced by children, by the internet, by the 50 peso note, by Minecraft, by an enormous popular following as a kind of permanent juvenile, an animal whose appeal lies precisely in the qualities the actual wild species has been refining for 15 million years. The cuteness is real. It is also, in a sense, the visible surface of something the popular framing rarely engages with. The fact that the animal almost everyone has met is a particular cultivated form of a species whose natural form most people have never seen, and whose natural habitat is being lost during the lifetime of the very people who keep the cultivated form in their bedrooms. Children growing up with pet axolotls in 2026 are growing up with a species whose wild population may not survive their childhood. Whether that gap registers in any wider cultural way is a question that conservation biologists discuss with each other carefully, because the answer is consequential and not yet known. What can be said is that the captive population, for all its abundance, exists because of the wild population that produced it. The 34 animals on the ship from Veracruz in 1864, the breeding colony in the converted monkey house, the slow distribution across European laboratories in the 1870s, the 1935 import to Buffalo, the 1962 cross with the Minnesota albino, the 2005 transfer to the Ambystoma Genetic Stock Center in Lexington, where the central reference colony for axolotl genetics now sits. Every one of those events was made possible by a wild population in the Xochimilco canals that, at the time, was healthy enough to spare animals. The captive population is, in the long view, a downstream consequence of a source population that has been failing for 40 years and may not be there much longer. If the wild population disappears, what remains will be the descendants of 160 years of laboratory breeding, with their narrow genetic base and their tank-shaped behaviors and their introgressed albinism and their cultural role as soft pink ambassadors of a species whose dark wild form will exist only in preserved museum specimens. They will still be axolotls. They will be a particular kind of axolotl. They will not be the animal that evolved in Xochimilco for 10 or 15 million years. The animal that evolved in Xochimilco will, at that point, be over. The animal in the dark canal, hovering above silt, is not aware of any of this. It does not know that its species is, in absolute numbers, doing very well. It does not know that the survival of the form it represents, the wild form, the dark olive-green form, the form selection actually produced, depends on whether a relatively small group of ecologists, agronomists, and Chinampa farmers in southern Mexico City can, against considerable pressure, keep its habitat alive long enough for the wild population to recover. It is just an animal in a place. The place is in trouble. The animal continues for now. On a clear morning in February of 2022, a small crowd gathered along the edge of one of the canals in Xochimilco for what had been organized with some ceremony as a release. Drummers and flute players in pre-Hispanic costume opened the gathering. Bowls of copal incense were lit, sending the resinous smoke that has accompanied Mexican religious and civic events for several thousand years drifting across the surface of the canal. Several officials from the local boroughs of Mexico City spoke briefly. Then, into the water, 6 captive-bred axolotls were placed, one at a time, by hand. The animals, leucistic, dark-eyed, pale pink against the green of the canal, slipped out of the cupped hands of the people releasing them and disappeared into the silt. The crowd applauded. The flutes resumed. The ceremony lasted perhaps an hour. The release was, in the most direct sense, symbolic. 6 animals do not save a species. The location, a publicly accessible stretch of canal rather than a fully prepared refuge, meant that the animals were entering an environment with the same tilapia, the same carp, the same water quality problems that had reduced the wild population in the first place. The biologists who had organized the event knew this. They had been clear about it in the planning. The release was not a recovery effort. It was an act of public attention, intended to draw the city's gaze, however briefly, to a wetland and a species that had been quietly disappearing while a megacity grew up around them. The actual conservation work, the slow, technical, unglamorous work that has any real chance of holding the species in the wild, was happening and is still happening somewhere else, on smaller stages, in the hands of a much smaller group of people. The person at the center of that group is a biologist named Luis Zambrano Gonzalez, who has been working on the Xochimilco wetland and its axolotls for more than 2 decades. Zambrano is a freshwater ecologist by training, based at the Institute of Biology at the National Autonomous University of Mexico, UNAM, and runs what is called the Laboratorio de Restauración Ecológica, the Laboratory of Ecological Restoration, which he founded in 2004. The laboratory's work, simplified to its core, is the attempt to keep enough of the original Xochimilco wetland functioning for long enough that the wild axolotl population has somewhere to recover into. Zambrano describes the problem in plain terms. We have to preserve Xochimilco in order to have axolotl, he has said in several interviews, comparing the relationship to the relationship between Arctic ice and polar bears. The animal cannot be saved without the place. The place is what produced the animal, what selected for the trait that made it biologically remarkable, and what continues, in its damaged form, to be the only environment in which the wild form of the species exists. The strategy his laboratory has developed is, in retrospect, almost obvious, and in practice extremely difficult. It begins with the Chinampa system, the same Aztec engineering system described earlier in this script, the rectangular agricultural platforms separated by canals that the indigenous farmers of the Valley of Mexico built over a period of several centuries, beginning around the year 1000 of the Common Era. A small number of these chinampas are still being farmed today by a community of perhaps a few hundred chinamperos, small-scale farmers who continue the traditional practices of growing flowers, vegetables and herbs on the platforms, using the canal water for irrigation and the lakebed mud for fertilization, and who have, in some cases, been doing this on the same family chinampa for several generations. The chinampa system, when farmed in the traditional manner, without industrial pesticides, without chemical fertilizers, with the canals kept open and the willow trees that anchor the platforms maintained, produces water of unusually high quality. It is, in effect, a 1,500-year-old wetland filtration system that happens to also be agricultural infrastructure. What Zambrano's laboratory has done is partner with the Chinamperos who still farm in the traditional manner to convert sections of canal into what they call refugios, refugees. The conversion is technical and slow. Semi-permeable barriers are installed at the entrance and exit of a chosen canal section, fine enough to exclude tilapia and carp, but coarse enough to allow water flow. The surrounding chinampas are committed, by agreement with the farmers, to pesticide-free and chemical fertilizer-free cultivation. The water inside the refuge is monitored for bacterial counts, for dissolved oxygen, for the presence of native and invasive species. As of late 2024, there are approximately 48 of these refuges distributed across roughly 5 linear kilometers of canal in the southern reaches of Xochimilco. They are small. They are vulnerable to the next major flood. They depend on continuing cooperation from individual farmers who have other pressures on their land. They are, with all those caveats, the only thing in the wetland that can plausibly be called recovering axolotl habitat. The animals that have been placed into them, partly through translocations of wild individuals from worse habitat, partly through the release of captive-bred animals from UNAM's own breeding facility, have, in the most rigorous study so far conducted, survived. A paper published in PLOS One in April of 2025, authored by a younger member of Zambrano's team named Alejandra Ramos, tracked 18 captive-bred axolotls released into either a restored Chinampa refuge or an artificial spring-fed wetland on the UNAM campus. Each animal carried a small radio transmitter. The team monitored their movements, their feeding, their use of available habitat for 40 days. All 18 survived the initial period. Most gained weight, which indicated successful hunting. Several formed what the researchers, with appropriate scientific caution, described as social associations, patterns of repeated proximity between specific individuals that did not appear to be random. 2 animals in the Chinampa refuge were eaten by great egrets, a wetland bird that hunts by spear strike from above, and that captive-bred axolotls, having never encountered a skyborne predator, apparently failed to recognize. The team is now developing what they are calling predator awareness training, a kind of pre-release conditioning protocol involving silhouettes and shadows passed overhead during the rearing period. Whether such training can be effective in an animal whose visual system is poor enough that it likely cannot resolve a flying egret in time to react is, in the team's own framing, an open question they are testing rather than a solved problem they are deploying. Running parallel to the refuge work is the population census. The most recent comprehensive count in Xochimilco, before the current effort, was conducted in 2014 and produced the figure of 36 animals per square kilometre that has been mentioned earlier. The survey now underway started in 2024 and being conducted across 2024 and 2025 is coordinated by Vania Mendoza, a biologist on Zambrano's team. It uses a combination of methods: traditional sampling, careful canal-by-canal walks, visual surveys in clear water sections, occasional capture and release of detected animals, is paired with environmental DNA sampling in which water is filtered through fine mesh and the genetic material left behind by passing animals is sequenced to determine which species have recently moved through. The eDNA method is well suited to a species this rare. It can detect the trace of an axolotl that swam through a section of canal hours or days earlier without anyone needing to see the animal directly. The census is ongoing as of this recording. Preliminary indications are that the wild population continues to decline. A modelling exercise based on current trends, published informally by the UNAM team in late 2023 and refined in 2024, projects that wild axolotls may be undetectable in the canals by the end of 2025. The team has been careful, in public statements, not to present this projection as a certainty. They have also been clear that they consider it more likely than not. There is a public-facing dimension to the work that operates alongside the technical one and is, in some ways, equally important. UNAM, in 2023, launched a program called Adopt Axolotl, Adopt an Axolotl, that allows individuals and institutions to fund the maintenance of captive-bred animals or contribute to the Chinampa Refuge program through small donations. The program has been supported by several Mexican companies, including, somewhat unexpectedly, the the financial subsidiary of the Latin American food delivery app Rappi, which now offers a co-branded credit card that allocates a fraction of its transaction fees to axolotl conservation. There is a museum, Axolotitlan, in Tarango Ecological Park in the south of Mexico City, dedicated to the species. The Senate of Mexico declared the 1st of February the National Day of the Axolotl in 2018, following the publication of the Nature Genome paper that year. The Bank of Mexico printed an axolotl on the back of the 50 peso banknote in 2021, alongside maize and chinampa imagery. None of these gestures in itself saves a species. Together, they construct the kind of cultural attention that conservation programs rely on for political support, and political support is, in this case, what the work needs to continue. The political support has not been steady. During the administration of Andrés Manuel López Obrador, which ran from 2018 to 2024, the budget of Mexico's Environmental Ministry, Semarnat, was significantly reduced as part of broader fiscal austerity. Conservation programs that depend on federal funding suffered accordingly. UNAM's work, fortunately, sits inside the university budget rather than the environmental ministries, but the wetlands regulatory protection, its enforcement against illegal construction, its water quality monitoring, the maintenance of the treatment plant at Cerro de la Estrella, relies on federal capacity, and that capacity has been thinner than the work requires. Whether the new administration that took office in late 2024 will restore the relevant budgets is, as of this recording, not yet clear. Zambrano, asked what he thinks the realistic outlook is, has been consistent in his framing. The captive population, he says, is an insurance policy. It exists for the case in which the wild population disappears entirely. It is not the goal. The goal is keeping the wild population alive in the place that produced it, because anything else means losing the species in the form selection actually shaped, and gaining, in exchange, a permanent laboratory population whose biology will continue to drift away from the original under the pressures of captivity. He has said, in interviews that tend to be quoted more than they are quoted accurately, that if the axolotl's population and habitat can be restored in a city of more than 20 million people, I feel that we have hope for humanity. The line is not exactly hopeful. It is more carefully balanced than that. The conditional is doing real work. The hope is conditional on the work succeeding, and the work succeeding is not certain. What is certain is that the work continues. The chinampas are being maintained. The refugees are being expanded. The census is being completed. Captive-bred animals are being raised, conditioned, in some cases released. The chinamperos who still farm in the traditional manner are continuing to farm. The animals in the refuges, at least the ones that have not been eaten by egrets, are gaining weight and, in at least some cases, breeding. The biologists at UNAM are publishing what they find. The cultural attention, helped by the 50 peso note and the Senate declaration and the museum and the Pokemon and the Minecraft and the Rappi credit card, is for the moment holding. It may not be enough. It may be enough for slightly longer than the 2025 projection suggests. It may, with a major change in the political support and a major investment in the wetlands water infrastructure, be enough to allow the wild population to begin recovering. The honest answer is that no one knows, including the people doing the work. They are doing it anyway, because the alternative is to do nothing. And nothing has been the trajectory for the previous 40 years, and is what produced the current situation. Doing the work is what gives the wild form of the species the chance, however small, of continuing. Somewhere in the canal system, in the cool water, perhaps in one of the refuges, and perhaps in a less protected stretch where the carp still dominate, an axolotl is hovering above silt. The number of axolotls that can do what this animal is doing in this particular place has fallen to between 50 and 1,000. Tomorrow morning, a member of Vanya Mendoza's team will collect a water sample from this section of canal and filter it through a fine mesh. Whatever genetic material the animal has shed into the water, a few skin cells, some mucus, a trace of metabolic exchange, will, 3 weeks from now, appear as a faint signal in a sequencing machine in a UNAM laboratory. The signal will say, in effect, something passed through here. The animal will, by then, have moved somewhere else, or it will not have. The work goes on either way. The question about human application has been hovering over this script for several chapters now, and it is worth answering as carefully as the science currently permits, because the question matters and because the popular versions of the answer are almost all wrong in roughly the same way. The popular version goes like this. The axolotl regenerates limbs. We have its genome. Its cells do something extraordinary. Therefore, with sufficient research investment, humans will, sometime soon, be able to regenerate limbs as well. This story appears in headlines reliably whenever a new piece of axolotl research is published. It is, in nearly every iteration, a misreading, sometimes a small misreading, sometimes a substantial one, of what the actual researchers have said. The careful version is more interesting and requires more patience to follow. What axolotl research is actually producing and what it will continue to produce over the coming decades is a slowly accumulating understanding of the regulatory conditions under which complex tissue regeneration becomes possible in vertebrate bodies. The genes are conserved, the toolkit is conserved. The difference between an animal that can rebuild a limb in adulthood and one that cannot is not the toolkit, but the regulatory state, the immune environment, the cellular plasticity, the signaling architecture, the absence of certain inhibitory cascades that mammalian bodies install during development and never remove. The translational question in this framing is not how to give human cells abilities they do not have. It is how to convince human cells in specific medical contexts to access abilities they already possess in attenuated form. The applications most likely to emerge from this line of work on a horizon of roughly the next 10 to 30 years are not whole limb regeneration. They are narrower, and in many cases they are already underway. Scarless wound healing, in which the cellular and immunological signals that produce mammalian fibrosis are modulated to mimic the resolution-favoring profile that axolotl macrophages run by default, is being investigated in mouse and rat models with promising early results. Fibrosis prevention more broadly, in heart tissue after infarction, in liver after injury, in lung after chronic damage, is one of the most active translational areas currently using axolotl-derived insights. Spinal cord injury, where the mammalian glial scar is the central obstacle to functional recovery, is another. An enzyme called chondroitinase, applied experimentally to mammalian spinal cord injuries, has been shown to degrade some of the inhibitory matrix that prevents axon regrowth, and the broader question of how to suppress the glial scar response while permitting controlled axon extension is one in which axolotl biology has been instructive. Cartilage and joint regeneration, where small defects in axolotls heal completely and equivalent defects in humans heal poorly, is a fourth. The work is not flashy. It does not produce headlines very often, because none of its individual steps look like science fiction. It looks like careful comparative cell biology, careful pharmacology, careful animal model studies, and careful clinical trial design, the slow industrial process by which translational medicine actually proceeds, and which produces, after long delays and many failures, the modest but real improvements in clinical practice that medicine accumulates decade by decade. A useful recent example comes from the laboratory of Prayag Murawala at the MDI Biological Laboratory in Maine. In work published in early 2025, Murawala's group showed that connective tissue cells, fibroblasts in particular, of the kind that carry positional information in their gene expression, are not merely contributors to the regenerative blastema, but are essential to it. When these cells are selectively ablated in axolotls before amputation, the blastema fails to form, and regeneration does not proceed. Other cell types, including the cells normally thought of as the more obvious progenitors, cannot substitute. The finding is technical, and its publication did not produce headlines. Its implication, however, is consequential. It means that any plausible program to enhance human regenerative capacity in a specific tissue will need to engage, specifically and directly, with the connective tissue compartment of that tissue, not muscle cells, not nerve cells. The fibroblasts, the cells most clinical researchers had previously regarded as supporting infrastructure rather than as protagonists of repair. Murawala, asked to summarize what the work means, has been clear in repeated public statements. The research does not provide an overnight solution, he has said, in essentially those terms. The advances will be incremental. The applications will be specific. The full translation, if it ever arrives, will look nothing like an axolotl. It will look like better outcomes after surgery, fewer scars, more complete nerve recovery, more durable joint repair. It will be, in the framing the field has consistently offered, less impressive than the popular version and more useful. The deeper teaching of the animal is not, in any case, primarily medical. It is something closer to a piece of conceptual rearrangement, the kind of finding that, once it is properly absorbed, makes you think differently about a category you previously considered settled. What the axolotl shows with unusual clarity is that the human body is not as thoroughly closed as our intuitions about it suggest. The genes that build a limb are the same genes that rebuild one. The cellular flexibility of an embryo is not, in any deep biological sense, gone from an adult. It is merely held in suppression by regulatory architecture that mammals install during development and never reverse. The capacity to regenerate complex tissue is not a feature only certain animals have. It is, in most respects, a feature most vertebrates have, held in different states of expression. Some animals, the axolotl most prominently among them, keep more of it open. Others, most mammals, including us, close most of it down. The closing is not absolute. There are tissues in the human body that retain regenerative capacity, sometimes substantial: the liver, the bone marrow, the epithelial lining of the gut. The question of why some tissues retain it and others do not is the question the field is now asking, with the axolotl as a kind of natural experiment in what an animal looks like when the closing has not happened. This rearrangement of how to think about regeneration is, in the long view, probably more important than any specific medical application. The applications will arrive in their narrow, careful forms on their own slow schedule. The conceptual shift, the recognition that the human body is, at the cellular level, a closed system that could in principle be partially opened, has already arrived and is already changing how researchers in regenerative medicine talk about what they are trying to do. They are no longer trying to give the body something it does not have. They are trying to permit the body to do, in specific narrow contexts, things it has the equipment for and is normally instructed not to do. There are, of course, things we still do not understand. The size of the axolotl genome, 32 billion base pairs, remains largely a mystery. Whether the bulk of it is functional, or whether it represents accumulated genomic real estate the species has tolerated rather than required, is unsettled. the encoding of positional memory in fibroblasts, how a cell knows it sits at the elbow rather than the wrist, and how that memory survives dedifferentiation and re-emerges to direct the construction of the correct limb segment, is being mapped but not yet understood. The relationship between captive and wild axolotl regenerative capacity is essentially unstudied. Whether the laboratory animals, with their narrow genetic base and their long history of artificial selection, retain the same regenerative profile as their wild ancestors is an open question that may become urgent if the wild population disappears. The mechanism that holds the metamorphic suppression in place at the level of the hypothalamus is partially worked out but incompletely. The reason the species refused metamorphosis in the first place, in the specific lake system where the refusal originally occurred, is plausibly explained but not directly demonstrated. The questions accumulate. They get answered slowly, in pieces, by laboratories scattered across several continents, in papers that appear in journals most people will never read. The cumulative effect of the answering over time is a slowly clarifying picture of what this animal is and what its biology has to offer to the broader project of understanding how vertebrate bodies work and how they could, in narrow specific contexts, be encouraged to work better. The animal itself, of course, has nothing to say about any of this. It does not know its genome has been sequenced. It does not know that a finding about its connective tissue cells in a main laboratory has implications for how reconstructive surgery may eventually be performed. It is just an animal. It has been doing what it does for somewhere on the order of 10 million years. The science is 1.5 centuries old. The questions the science can now begin to answer were unaskable in any precise form until the last 20 years. The questions the science cannot yet answer will, in some cases, become answerable in the next 20. In others, they will not. The animal will continue, under whatever conditions remain available to it, doing what it has always done, indifferent to the conceptual rearrangements it is producing in the species that has been studying it. The wild population, on the most current evidence, sits somewhere between 50 and 1,000 individuals. The estimate is wide because the animals are genuinely difficult to count. They are dark, they hold still, they live in water that is not visually clear, and they exist in a wetland that has been carved up by canals and chinampas in ways that make systematic survey work slow and expensive. The ongoing census being conducted by Vania Mendoza's team will, when it is complete, sharpen the number. The trajectory the number sits inside, however, is not in serious dispute. Between 1998 and 2014, the wild population in Xochimilco lost roughly 99% of itself. Since 2014, the trend has continued downward. The modeling exercise published by the National Autonomous University of Mexico in late 2023 and refined in 2024 projects that wild axolotls may be undetectable in the canals by the end of 2025. The model could be wrong. The trend could plateau. A major restoration of the wetland's water quality, paired with a meaningful suppression of the invasive carp and tilapia populations, could give the wild animals enough room to begin recovering. None of these outcomes is the most likely one, on current evidence. The most likely outcome is that the wild form of the species, the dark olive-green animal with the gold flecks along its spine, the form that 10 or 15 million years of selection in the lake systems of central Mexico actually produced, will be functionally extinct in the wild within the lifetimes of people now reading this script. What will remain, if that happens, is the captive population, somewhere in the hundreds of thousands, distributed across laboratories and pet shops and bedrooms on every inhabited continent, descended from the 34 animals that crossed the Atlantic on a French ship in 1864, carrying introgressed alleles from a Minnesota tiger salamander hybridized into the lineage in 1962, selectively bred for traits convenient to laboratory work and to the pet trade. These animals will still be axolotls. They will be a particular cultivated form of the species, useful for research, charming as companions, increasingly genetically narrow, behaviorally adapted to tank life rather than to wetland life. They are not, in any straightforward way, a substitute for the population that produced them. They are what is left when the source has gone. Whether the source has to go is a different question. Luis Zambrano and his team, the Chinamperos who still farm in the traditional manner, the small constellation of biologists and ecologists and government officials who have managed to keep the Chinampa refuge program alive through budget cuts and political turbulence, these people have not yet given up. They have a clear understanding of how difficult the work is. They are doing it anyway. Whether the work succeeds depends on factors most of them cannot control: federal funding, water treatment infrastructure, the political will to enforce wetland protection against the constant pressure of urban expansion, and the slow, patient cooperation of farmers who could, if they chose, walk away from the traditional Chinampa system tomorrow and replace it with something more profitable. None of those factors is guaranteed. Several of them have been deteriorating for years. The work continues anyway, because the alternative is not doing it, and not doing it is what produced the current situation. There is a longer view, and it is worth taking before this script ends, because the longer view is what this animal has spent the entire script asking for. The axolotl, in its wild form, is the product of a decision its lineage made somewhere in the deep evolutionary past. A decision encoded not in any conscious choice the animals ever made, of course, but in the slow accumulation of selection pressures favoring individuals that did not metamorphose, in a lake system stable enough that staying in the water became the better option than crossing to land. This trajectory has been running in the central Mexican highlands for somewhere on the order of 10 million years. During that time, the animal has been, simultaneously, an ordinary salamander quietly hunting invertebrates in shallow lake-edge habitat, and a creature carrying inside its body the most thoroughly preserved regenerative program of any complex vertebrate on Earth. The two things are the same thing. The regenerative capacity is not a separate gift bolted onto an otherwise unremarkable animal. It is the consequence of the developmental decision the lineage made and would have been impossible without it. To understand the axolotl is to understand that maturity in vertebrates is a closing, that adulthood, in our lineage and in most others, is the moment the developmental options seal, and that this single species, alone among its complex-bodied peers, declined to perform the sealing. For most of the time the lineage has existed, no one was watching. The lakes of the Valley of Mexico were uninhabited by humans for the first 9.5 million years of the animal's evolutionary history. When humans did arrive, beginning around 8,000 years ago, they noticed the animal almost immediately. The middens of the early lake-edge settlers contain its bones. The Aztecs, arriving in the basin in the 14th century, watched it for 2 centuries before the Spanish came and built it into their religious imagination so thoroughly that it carries today the name of one of their gods, Xolotl, the dark twin of Quetzalcoatl, the deity who refused his own sacrifice and fled into the water rather than complete the transformation expected of him, and who chose as his final form this strange small creature in the canal. The myth, as a piece of biology, is uncomfortably close to accurate. Whether the Aztec naturalists who built the story noticed something the science would not catch up to for 400 years, or whether the alignment is the product of a coincidence so clean it strains the word, is something this script has not been able to settle and probably cannot be settled by anyone. What the alignment makes clear is that this animal has been present in human imagination with surprising precision for as long as humans have been present in its valley. What it asks of us, to whatever degree an animal can be said to ask anything, is not that we save it for sentimental reasons, or that we extract from its biology a cure for human conditions, or that we make of it a symbol for any of the things humans habitually make of unusual animals. What it asks of us is that we recognize what kind of biological event it represents. 10 or 15 million years of selection in a small high-altitude lake system produced a vertebrate that does with its own body what no other vertebrate of comparable complexity has been observed to do. The conditions that produced it, the stable water, the abundant food, the absence of severe predators, the long uninterrupted span of evolutionary time, exist nowhere else on Earth. If the wild population disappears, the biological event that produced this animal does not get replayed. The captive population continues, useful and abundant, but the experiment that produced it ends, and the conditions for any equivalent experiment ever happening again in this species or any other are vanishingly unlikely to recur. The lakes are mostly drained. The pressure is mostly urban. The window in which an animal like this could come into existence has, for practical purposes, closed. This is the deeper teaching, if the word is not too grand. The axolotl is not just an animal that can regrow its limbs. It is a piece of biological evidence about what evolution can do, given the right conditions and enough time, that we are in the process of losing, not because the animal is failing, but because the conditions that sustain it are. The captive population preserves the genome and the form. It does not preserve the experiment. The experiment requires the lake. In the canal, sometime in the small hours of the morning, an axolotl is hovering above silt. The water around it is a few degrees cooler than the air above the surface. Its lateral line is registering the slow current. Its ampullary organs are reading the electrical state of the wetland around it, the heartbeat of a buried crustacean, the firing nerves of a small fish moving past, the standing field of the city overhead. 22 million people going about whatever they go about at this hour. The animal does not know any of what has been described in this script. It does not know that its species has been a god and a meal and a model organism and an internet phenomenon and a 50-peso note. It does not know that it carries in its cells the most studied regenerative program in vertebrate biology. It does not know that it has been a child for 10 million years, and that being a child is what allows its body to do what its body does. It does not know that its kind in the wild may not last out the decade. It is just an animal in a place. The place is dark and cool and full of the small, slow movements of other living things. The animal continues, paying its quiet attention, the way its species has been paying attention here since long before there were people in the valley to notice. Good night.